in respect to the speakers, and also a gentle reminder to the poster presenters. If you haven't already, please put up your posters on the boards outside. Thank you. Okay, a very good morning to professors, associate professors, invited speakers, intrigued guests, ladies and gentlemen. My name is Siralan. I'm from first year medical student, and it is my pleasure to meet you all and be your MC this morning. First and foremost, I would like to thank MAHSA University for organizing this event and the co-organizer, which is Vivekananda College of Arts and Science. On behalf of the school, on behalf of the School of Medicine, MAHSA University, I would like to extend a warm welcome to Yang Berhormat Professor Dr. Rusdi Dimodin, Chairman of MyMed 2023, Dean of Faculty of Medicine, MAHSA University. Yang Berbahagia Professor Dr. Ikram Shah bin Ismail, Vice Chancellor of MAHSA University. And last but not least, Yang Berhormat Professor Dr. Rosna binti Muhammad Zaini, Deputy Vice Chancellor, Research, Innovation and Postgraduate Studies of MAHSA University, and fellow Deputy Deans, lecturers, invited speakers, and fellow attendees. We would now like to start with a Dua recitation by Dr. Muhammad Fakhrul Razi bin Haji Muhammad Saleh.
Bismillahirrahmanirrahim. Alhamdulillahi Rabbil Alamin. Wassalatu wassalamu ala ashrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Alhamdulillahi Rabbil Alamin. Hamdan yuwafi ni'amahu wa yukafi mazidah. Ya Rabbana lakal hamdu kama yanbaghi li jalali wajhikal azim sultanik. Allahumma salli wa sallim ala sayyidina Muhammadin salatan tunjina min jamil ahwali wal afat wa taqdi lana biha جميل الحاجات وتطهرنا بها من جميل السيئات وترفعنا بها عندك على الدرجات وتبلغنا بها الأقصى الغايات من جميل الخيرات في الحياة وبعد الممات. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار وادخلنا الجنة مع الأبرار يا عزيز يا غفار يا رب العالمين. وصلى الله على سيدنا محمد وعلى آله وصحبه أجمعين. الحمد لله رب العالمين.
Okay, thank you so much, Dr. Razi, for the dua, and I hope this event will go well today. Now, I would like everyone to please rise as we play our national anthem, Negaraku, followed by our MAHSA University song.
Menggapai cita, mengutarakan impian. Kita sang juara, mengeraju cemerlang. Selaluan keinginan, aspirasi negara dijulang. Bersamakan keyakinan seluruh wadah, kita megahkan jaringan antara bangsa. Disinilah dengan nasional berjaya, kita lakukan di pentas dunia. Universiti MAHSA, lambang kejayaan. Universiti MAHSA, satu kemegahan. Universiti MAHSA, menuju kemasyhuran. Universiti MAHSA, Universiti pilihan. Berdaya saing, berinovasi, berdedikasi, berintegriti, melahirkan insan berkualiti. Bangsa jadikan satu yang ini.
Okay, thank you so much. Please sit down. Okay, to begin, let's have a warm welcome on stage, Yang Berhormat Professor Dr. Rusli bin Nodi, to deliver the welcoming address.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh. A very good morning. Thank you, Mr. Chairman. Yang berbahagia Profesor Dr. Ikram Shah, Vice Chancellor. Yang berbahagia Profesor Rosnah Zain, our Deputy Vice Chancellor. Distinguished guests, Deputy Deans, Dr. Anthony Samrod, Chairman of the Organizing Committee, and members, all the delegates, all students, welcome to this MAHMAT 2023. So on behalf of MAHSA University, I'm very honored today to be here with you today to welcome you to this conference. And this is again a truly exciting time for all our Faculty of Medicine as we gather to discuss latest advances, medical research and practice. Although we are having this conference, just to note that during this time, we still have to remember one of our colleagues is in hospital, in critical care. So I think while I'm reading this, perhaps we can still have a prayer for Dr. Sun Suu Chu, who's struggling in critical care for surgery. So just to share that very sad news, hopefully that God will help her, God willing that she will not survive and also come back to us. So again, on this note, I would like to thank our sponsors for their generous support for this conference and your contribution, all of you, not only one or two, but all of you, okay, are essential to our ability to bring together a distinguished group of national, international speakers and attendees. I would like to thank all our speakers. I think without you, I think this conference will not achieve its aim. So all speakers, all presenters, oral and posters, right? And for your expertise, your presentation will help us to stay ahead of the curve in terms of medical knowledge and practices. The theme of this year's conference is multidisciplinary approaches for sustainable health. This is a timely topic as the field of medicine is constantly evolving and we are facing new challenges such as the rise of antibiotic resistance and the increasing prevalence of chronic diseases. However, we are also making great progress in areas such as gene therapy and personalized medicine. Thus, we are at a critical juncture in the history of medicine. But we have the opportunity to make significant advances in the coming years, and that could be done if we would work together. I know that many of you have traveled long distances to Malaysia and to MAHSA University, and I want to thank all of you for your commitment, for your presence, and together we can make this a great conference. So we are gathered here today because we are all passionate about our medical, nursing, and life sciences. We are all committed to make a difference in the world, and I believe that this conference will give us the opportunity to move ahead. I think that this conference will provide us with a forum to discuss these challenges and opportunities. I'm confident that we will leave here with a better understanding and awareness of the future of medicine and how we can shape it, how we can manage it. So in closing, I would like to thank again all of you for being present today in MAHSA University, one of the top private university in Malaysia, and I wish you a productive, enjoyable conference. Thank you very much.
Thank you so much. Thank you so much, Professor Dr. Rosli. Next, let's welcome on stage Yang Berbahagia Professor Dr. Ikram Shah bin Ismail to deliver the opening remarks.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and good morning, and thank you, Chairman. Yang berbahagia Profesor Rosnah Mazin, Deputy Vice Chancellor, and the Dean, Professor Rosli, and our new YB. Belum election ada Pak YB dah. Pak Rosli, Dean of Faculty of Medicine, Deputy Deans, academic staff, and also special welcome to all the students from India, Satya Bharma and Vivekananda. Where are you? Oh, good. Nice to see you. So you travel all over Malaysia now? Or not yet? Thank you. Thank you for being here today as we gather for this conference, MAHMAT 2023, where we have the opportunity to exchange thoughts, ideas, and innovations that will shape our future. It is truly an honor to stand before such a brilliant group of individuals who are dedicated in making a positive impact in the world. In this age of rapid technological advancements and globalization, we find ourselves constantly adapting and evolving. The challenges we face are vast, from climate change to economic inequality to social unrest. However, it is in times like these that we must come together to find solutions that build a sustainable and harmonious future for all. To foster collaboration, we must build bridges of understanding and empathy. When we are open to diverse perspectives and are willing to listen and learn from one another, that is when true progress emerges. The old adage, two heads are better than one, rings true in this context. When we come together, pooling our knowledge and expertise, we are able to find innovative solutions that surpass anything we could achieve individually. In the field of medicine and life sciences, our impact on humanity is tremendous. Every day, doctors, scientists, researchers, and caregivers work tirelessly to improve the quality of life for individuals and communities. From developing new treatments and therapies, to uncovering the mysteries of the human body, to promoting preventive care, the advancements in medicine and life sciences have saved countless lives and improved the well-being of millions. But our work is far from over. There are still diseases to be cured, inequalities to be addressed, and innovations to be discovered. That is why conferences like these are so important. They serve as a platform for the exchange of knowledge, forging of partnerships, and the inspiration for new ideas. So at the course of this conference, I encourage you to engage in meaningful conversations, to challenge existing paradigms, and to explore new avenues. Let us come together in a spirit of collaboration, innovation, and a shared vision of a brighter future. Together, we can make a real difference in the life of individuals and communities across the globe. Thank you once again for your presence here today. I have no doubt that this conference will be a catalyst for positive change, and I look forward to the transformative discussions. Thank you.
Thank you so much, Professor Dr. Ikram. Last but not least, let's welcome Yang Berhormat, Professor Dr. Rosna binti Muhammad Zain, to deliver the special remarks.
Bismillahirrahmanirrahim. Assalamualaikum and a very good morning to all. Yang berbahagia, Professor Dr. Ikram, Vice Chancellor, MAHSA University, Professor Dr. Rusli, Dean, Faculty of Medicine, Dr. Anthony, and all distinguished guests and participants. I'm honored to be here with you today in this MyMed 2023 conference, where we come together to celebrate the relentless pursuit of knowledge and innovation. It is indeed an honor to have you all gathered here today as we embark on a journey to explore innovative and collaborative strategies to achieve a healthier and sustainable world. I would like to start by expressing my heartfelt gratitude to each of you for joining us at this significant event. Your presence exemplifies your commitment to making a positive impact on global health and well-being through multidisciplinary collaboration. I would also like to convey my special thanks to the keynote speakers and oral and poster presenters who are here to share their knowledge and expertise with us. The theme of this conference, Multidisciplinary Approaches for Sustainable Health, is not just a catchy title, but represents our collective determination to address the challenges of today and tomorrow. By synergizing our diverse expertise, knowledge, and insights, we aspire to lay the groundwork for a sustainable and healthier future for generations to come. This gathering provides a unique opportunity for experts, scholars, and professionals from various disciplines to come together under one roof. It is at this intersection of specialties that we can foster new ideas, innovative solutions, and groundbreaking research, all of which will lead us towards a more sustainable and resilient healthcare ecosystem. Throughout the conference, we encourage you all to actively engage in knowledge exchange and collaboration. Share your insights, experiences, and research findings openly, for it is through this intellectual dialogue that we can unlock the potential of true multidisciplinary approaches. Beyond the scheduled sessions, take advantage of the networking opportunities this conference offers. Forge new partnerships, build bridges between disciplines, and establish lasting connections that will strengthen our collective efforts in the pursuit of sustainable health. As we immerse ourselves in the enlightening discussion and interactive workshops, let us keep in mind that our mission extends beyond this conference. We carry the responsibility to apply the knowledge gained here to our respective fields and communities, contributing to a healthier and more sustainable world. Once again, I express my deepest appreciation to all speakers, sponsors, and attendees who have made this conference possible. Your commitment to driving change inspires all of us. Let us seize this moment to shape a brighter, more sustainable future for global health, one that embraces the power of multidisciplinary approaches. Thank you again, and let's make this conference an unforgettable milestone in the journey towards sustainable health. Thank you.
Thank you so much, Professor Dr. Rosna. Before we move on, I would like the oral presenters, please send in your soft copies of your presentations at the registry outside. Thank you. Okay, now let us move on to our keynote speech one. I would like to invite our chairperson, Associate Professor Dr. Masila Mani, to chair the introduction of the keynote speaker.
Very good morning, all of you. I'm Dr. Masila Mani, Associate Professor at Mama University, standing before you to introduce our first keynote speaker, Dr. Vikas Mehdi. So, coming to Dr. Vikas Mehdi, is a professor and he is an editor-in-chief, Indian Journal of Pharmacology. He is also editor-in-chief, International Journal of Pharmaceutical Sciences and Nanotechnology, and he is coordinator, PG IMER, Department of Pharmacology, Chandigarh, India. And regarding his achievement, his area of expertise is experimental pharmacology, clinical research, regulatory pharmacology, development of nanoformulations, pharmacogenetics, pharmacogenomics, and stem cells, etc. He has more than 20 of various funding agencies, Government of India, as well as international bodies, such as ICMR, DBT, DST, CSIR, NBA, MCI, etc., etc. Professor Mehdi has been selected as 2% of scientists across the world conducted by Elsevier and Stanford University. So, high profile person. We have here, he has authored 5 books and 160 book chapters and published more than 500 articles. It's a remarkable one in national as well as international journals. As I mentioned, Professor Vikas Mehdi is a member of various task force of India, almost all the funding agencies. It's excellent contribution towards science and research. And he is also a chairman for vaccine group of ICMR, member of Central Ethics Committee, ICMR, part of COVID-19 task force and antiviral screening committee, ICMR, etc. So with this small introduction, now I invite Professor Dr. Vikas Methi to share his keynote speech with us all. Welcome, sir. Thank you.
Thank you, sir. And Honorable Vice Chancellor, Dean, and all the dignitaries. So this is my second time. I came last February here. So what I wanted to talk about, like we have already experienced during the COVID time, how in a short period you can go ahead with a molecule. But conventionally, when you talk about from identifying from in silico to clinical development and approval process, I'll just give a fair background. So when you look at that, how do you develop a drug? Long time back, when you think that you wanted to develop a drug, even if you do not know mechanism of action, still you can go ahead. At least you can know that this has some effect. Couple of years back, we did not have any known mechanism of digoxin, but it was in the market. But today you go to the regulator and say that I don't know this part, and they say go back, do the study, come back to us. So that means each and every information you need to have today, how the drug is absorbed, how it is metabolized, how it is distributed and metabolized and excreted. You need to know. And if it is converted to active metabolite, how it is going to happen. So we have a branch of metabonomics now in order to deal with that. So in subsequently, if you say, take an example of diacerin, this is only one drug which is used for disease modifying effect in osteoarthritis. And now, when it was approved, people are still working on its metabolite orsin because this is an active metabolite. So I'll just give you a background. How do you go ahead? Can you change the slide? It's not working. Okay. This is what the real scenario is. You work on 10,000 molecules, and by the time you identify a hit and miss molecule, so that means the failure is very, very high. And coming to the cost factors, if you see how much money is required, it's a billions of report. Though you are thinking that we have all medicine for all the disease, and because we have a lot of students, I just wanted to highlight there are so many diseases we do not have a medicine, not a single medicine. And it came to my mind when we came to Singapore. And usually, whenever they conduct a meeting at conferences, they also call the people, patient. One of these meetings, they call people suffering from epilepsy. It's not to show that what kind of disease it is, wanted to show the researcher that how they should motivate it and patients should be motivated. I remember one of the case where one lady was called from Paris, and she is 5 gold medalist, and she is suffering from drug-resistant epilepsy. Similarly, we have an organization called PharmaXcel. If we say that India, we are the second largest for pharmaceutical manufacturer, we export the medicine to almost 200 countries, and you could see that during COVID time, when on 22nd March, because I was working on a task force for COVID, so we are deciding on hydroxychloroquine for prophylaxis. And that day, I only put the question that, do you have a hydroxychloroquine? Because we are 140 crore people. If 1% develop, 1% develop COVID, can we supply the hydroxychloroquine? And within 15 days, we could supply to 150 countries. and that's capacity to have in a country. Second thing is when you talk about any of the vaccine, we could supply to each and every country because that is why the Covishield also came to India because we have the largest manufacturer. But otherwise, if you see that talking about any disease, do you have any medicine for each and every disease? Answer is no. You can take an example of celiac disease. What do? Patient come to the OPD, we diagnose, and we only offer gluten-free diet. So you can make a list of diseases where we do not have a medicine, and this is for a student. Rare disease, I'll tell you because I'm a part of regulators. When we decide and approve any medicine, we are not supposed to ask very, very costly, which a common man cannot afford it. So can we develop an affordable medicine? And that is why it is very, very costly and still 50%, like you have to go to the extreme rural area, how much medicine is available. We talk about rationalized medicine, essential medicine concept. That is why we do it. So if you look at that, how it is developed, like if you say, initially, you identify receptors. If you have a receptors, then you say that this is what the pathophysiology is. Then you can see that can I develop a molecule? So you do a simulation technique. Okay. So in this simulation technique, this in silico data you develop. Can this particular product, like in COVID, what we did, we wanted to develop a drug which go for emergency approval within a couple of days. So we check in silico method by 75,000. We have 78,000 molecules in the zinc database and other database. So we just say that which molecule will be working. So we have an experience of Ebola virus, okay, when you look at the COVID. So can it act as, in a COVID virus? So we identify the target, N-terminal, M-terminal, okay, S1, S2. And that's how we try to repurpose all these drugs. If you take an example of COVID, like hydroxychloroquine, so CDC had already found that chloroquine is also acting, hydroxychloroquine is also active. It is more effective in COVID. But why we had gone for a hydroxychloroquine? When you compare chloroquine and hydroxychloroquine, there are more accumulation in a drug concentration on fifth day with hydroxychloroquine. Because initially in the first wave, we thought like we have a concept that COVID only affect the lung tissue. Later, we came to know that it is a multisystemic disease. And that's how on fifth day, high concentration is there. Half-life is 54 days. We go ahead with a hydroxychloroquine. And that is why that when you do in silico studies, so definitely what you go for proof of concept studies. So proof of concept study, you try to generate the mechanism of action. Okay, how it is acting, what dose it is acting. Then you decided to go for a regulatory toxicity study. So in preclinical, depending on clinical use, I'll talk about a little more that you wanted to use the drug for 10 days. What kind of data is required? You required only acute toxicity study. If you go beyond 30 days, what kind of data you require? You require a repeated dose toxicity study. That means if you use that drug for 10% of the lifespan, okay, you need repeated dose toxicity study, that is 90 days, 180 days, 1 year. Okay, you wanted to use the drug like for hypertension, for Alzheimer's, chronic disease, more than 6 months. If you go beyond more than 6 months, your question should be asked that, can it cause mutagenicity? Can it cause carcinogenicity? And that is why you need a long-term toxicity study that is for 2 years. But if you see that today we have a lot of product, like nano product, even if we use for 10, 15 days, still it is required mutagenicity and carcinogenicity because this product has capacity to retain in the tissue for a long time. And that is why you need to go for all this. Now, over a period of time, you might have seen that a lot of drugs have been withdrawn from the market. You can take an example, very popular like terfenadine, antihistaminic. It has been withdrawn. Cisapride, it is used in gastric dyspepsia. It has been withdrawn. It's not that drug is banned. It is because of it causes cardiac arrhythmia. And that is why there is a big branch has come up, which is safety pharmacology. Even if you today use a drug for any other condition, then you say, can it affect CNS? Can it affect respiratory system? So that's how you do a lot of safety pharmacology studies to find out that is it affecting other tissues. Similarly, if you go beyond the toxicity study, you could say that I wanted to do a phase 1 study. It is a healthy male and female volunteers. But tomorrow, after a couple of years, that male volunteer come back and tell you that I was not married when I was participating in the clinical trial. But I got married, I don't have a children. Okay? So that means maybe something wrong with the drug, I don't have a children. And that is why you do all reproductive toxicity studies, male and female reproductive studies. So that's how you go. So once you do that, basically you wanted to see that when you do acute toxicity study, repeated dose or long-term toxicity study, what dose can be used for a human? How safe it is? Okay? So you take out this particular dose and use in a human, and then you go for a phase 1, phase 2, phase 3, and phase 4, like that you go. So when you work on 5,000 to 10,000 molecules, ultimately you select maybe 1 and 2 molecules which will be suitable for human studies. And that you call it hit compound. That means you know some effect is there. When you call it a lead compound, you establish a structure-activity relationship and go with your patent. So you might have heard about generic medicine and branded medicine. Moment you go for a patent, you get exclusivity for 20 years. So that means 5 years you span on preclinical toxicity, then another 5 years is for clinical development. So by the time molecule come to the market, it is 10 to 12 years. So earlier it used to take 20 years because you did not have that technology to screen the molecule, high put through, ultra high put through. And that is why once you do that, you file an IND. IND is investigational new drug. That means you seek an approval that you wanted to do a human studies, phase 1 study. Okay, and that's how. So once you complete phase 3 study, you call it NDA, new drug application. So you seek the approval for marketing approval. Next slide. So here the clinical situation is there. You know that how the concept is changed because who you wanted to, if you happen to go to any Western countries, it's now concept is accelerated drug development process. Okay, because it takes earlier 20 years, now at least 12 years. Can you go for the accelerated development? So they said, don't do the preclinical study. Take a minute amount of drug, give it to the human. And that was the concept of microdosing or phase 0 studies. But it was not actually accepted for all over the world because we do not have that high put-through clinic, like very, very sensitive technique to establish the PKPD. And that is why conventionally what you do is once you do preclinical studies, you go for a human, healthy human volunteer. So you take around, maybe 20 to 80 depending on the molecule. You give that dose, then you establish a maximum tolerated dose. So from preclinical study, what you get it, you get, dose is, NOEL, no adverse effect at that level, or NOEL or LOEL, low adverse effect. So from that you extrapolate. Okay. Now the extrapolation is based on PKPD modeling. Once you do that, then you go to patient, phase 2. You do it in patient, how it is going to affect, you evaluate the safety and efficacy. So phase 2 also there are 2 parts. One is phase 2A and another is phase 2B. Phase 2A means you give it in a homogeneous population. Phase 2B is heterogeneous population. There are a lot of comorbid conditions, how drug is going to affect. Then you go for a phase 3 in a larger population, and here also phase 3A and phase 3B, homogeneous and heterogeneous population. Then you confirm this, whatever finding you get, you confirm it. Then you file, go to regulator, and apply for an IND. So normally in the US FDA, when you make an application, if you do not get any reply within 30 days, that's a good news. That means application has been approved. So even Indian regulator also applied, we have a digitalized one, Sugam portal. So when you make an application, if you do not get any reply, that's a positive one. You can go ahead with a human trial. Next slide. So this is what the process is. Sometimes, though I said 30 days, but if there is a queries, it might take time. So once you go, so finally one drug is approved. You could work on 5,000 to 10,000 compound. Next slide, please. So this is what the process is in summary. You identify a molecule, you go for in silico, proof of concept, then you go all regulatory toxicity studies, and then you do phase 1, phase 2, phase 3 study, but we have a provision of accelerated development. And that you could see in COVID. We approved more than 160 compounds. Based on what? In silico data, in vitro data. Based on what? It is trying to repurpose the data, looking at can it effective in Ebola? It is similar to that. Can it affect on S1, S2, M2, and N-terminal? Okay, so we have accelerated development. So this provision in pandemic situation, it is there. Next slide, please. So when you do preclinical, if you see that you work on number of compound, but success story is maybe 1 and 2. And that's a reality. That's a reality. So why do you do preclinical toxicity study? Because you know that now we talk about individualized variation. You might have seen that during the initial address, we talked about individual variation, precision medicine, personalized medicine. Okay, maybe you can think of that what you do in the preclinical. So can you identify some hazard and which can be translated into the human. Next slide, please. So if you look at the story, I'll just tell you the story that nowadays US FDA had recommended that you need not have to go for preclinical studies. You go for a cell-based. Even Indian regulator also recommended for some of the cell-based studies. Now, if you look at that, you read the story of thalidomide. Have you heard of drug named thalidomide? Anyone? So when you go and read the story, thalidomide was approved in 1957 by a German regulator. And during that time, regulator was not stringent. So what they did is they did a study in mice and rat, and they found it is to be very, very useful. But they also did a study in the rabbits. Rabbits are the mammal. And what they could find rabbit, they found it a teratogenicity. So what they did is they hide the result of the rabbit, and they went to regulator with rat and mice. And regulator approved it. Now, subsequently, when the drug was approved, one of these pediatricians, he was practicing pediatrician, and he had written a letter to editor to Lancet. And he write it like that. I'm a pediatrician. Normally, I see a congenital anomaly is 1.2%. But recently, I have seen a lot of phacomelia cases, children. Okay, so that became a signal. Then later, people came to know that there are 10,000 babies born with phacomelia. 10,000 babies. And this company also applied to US. And you happen to go to Maryland, where US FDA headquarters is there. There is a lady inspector. She was evaluating this file, and she found something wrong is there. She never approved this drug. And that's the reason there is not a single case in US. And later, this lady inspector was awarded by President of US. I also look up to the sports. When the Brazil Olympic is there, I saw a phacomelia case. Moment I say, is it because of thalidomide? His mother was there. Though they speak only Portuguese, said, yes, this is because of thalidomide. And that's a scenario. So when people went into court, the court has asked for all the data. They could find, they already knew that it causes teratogenicity. And that is what we learned, that there is a species variation. And that is why you say that any preclinical study you wanted to do, how it is affecting a rat and mice, smaller animal, you do it in higher monkeys and dog also. That is the basis. Second thing is that earlier, when you plan for any phase 1 study, people only say you are going to include only male healthy volunteers. Nobody wanted to do the studies in the female. Then the concept is coming is if you do not do the studies in female, you do not know how the drug is going to behave in female. So 1994 Canadian regulator, they come up with a concept, we must include both male and female. Okay, provided you have a protection for adequate protection for pregnancy. And that's how you see that in experimental also we do male and female. You want to see that, many of the drug we say that preferably it should not be used lesser than 12 years because we do not have enough. Okay, so that's the reason most of this pediatric population, whatever you use is off-label use. So that means you have been prescribing in adult for a couple of years, maybe 10 years, 15 years. Then there are off-label studies have been done. People use in a particular dose and found useful, then started using in pediatrics. So this is a scenario that you use all the drug in the preclinical, but please remember that whatever study you do, there is a concept called OECD. In experimental study, when you do, it is called, you have to do under principle of OECD. OECD is Organization of Economic Cooperation and Development. So they have a test guideline. So if you do not do under OECD, all preclinical study, regulator will not accept a study. And fortunately, we are very proud that India is going to lead OECD for next 3 years. Like we have a president for G20. So we are going to lead that OECD for next 3 years. Any clinical studies, we have to follow the ICH. ICH is earlier called International Council. Now it is called Council of Harmonization, earlier called Conference. So any study you say that I have to follow OECD guideline for preclinical, for clinical study like phase 1, phase 2, phase 3, phase 4, you have to follow the ICH guideline. So this is how. So when you plan for a study, how the concept of OECD came, you just read the story. There is a lab in Chicago. They will do like a lot of studies. So there is a complaint to Environmental Protection Agency and US FDA. And they started investigating in 1972. So by the time they investigated, they could already found that they have completed 22,000 studies. And when you look at these studies, they could find there a lot of falsification. Okay, and that's the reason they implement OECD principle that if you do OECD, the concept is I can go to Malaysia, I can go to US, I can go to Europe, anywhere I can do. It is accepted. If I do clinical study, I can go to across the globe, it is accepted. And that's the principle is there. Okay, next. So concept of doing an acute toxicity study, repeated dose toxicity study, chronic toxicity study. If we use for 10 to 15 days, only required is acute toxicity study. I'll just give you one example. COVID time, we might have used paracetamol so much. You know, there's a media report in India also, how much paracetamol was consumed. Now, if you look at the paracetamol, you know that in fever, we use 500 milligram 4 times in a day, maybe for 5 days. Okay, but how do you get to know that paracetamol causes a toxic effect if you go beyond 10 grams? Person may die. Okay, if you go beyond 10 grams or more than 10 grams. So this comes from acute toxicity study. So acute toxicity study will give you that what dose is suitable, what dose is poison. So it also give a direction from initial part that if you use beyond this, it is going to cause poisoning. How can you manage this patient? Similarly, for repeated dose toxicity study, that means if you use the drug for 10% of the lifespan, how the body is going to behave. And chronic one, if you use beyond 6 months. Any drug clinically used beyond 6 months, you require a long-term toxicity study. Because beyond 6 months, one question you might ask that, can it cause mutagenicity? Can it cause carcinogenicity? Okay, so that is why you need to find out. So one important point is many of the drug, it can cause mutagenicity, it may not develop carcinogenicity. Next slide. So that's how you go for that. You know, identify a target, then how do you go synthesis of chemical, lead optimization, candidate selection, do preclinical study, file an IND, then you go for all phase 1, phase 2, phase 3, phase 4, and ultimately you go for NDA application. So what does it mean IND? IND does not mean you are asking for a marketing approval. What you are asking is that you wanted to go for that, you know, first time in a human. So next slide. So this is what next slide. So when you make an IND application, the regulator requires certain data. What are the data? Have you done acute toxicity study, repeated dose, safety pharmacology, pharmacodynamics, genotoxicity, everything. So if you look at that reproductive toxicity, I'll just give you the background. No drug we approve during the pregnancy. So if you approve a drug for during a pregnancy, you need to know that segment 1 study, segment 2 studies, segment 3 study. What is a segment 1 study? Can it affect the implantation? There are a lot of drugs, it causes recurrent pregnancy. Okay? Can it affect the implantation? Can it affect during the period of organogenesis? During the reproductive, that is an example of thalidomide. Then, can the drug, when you give it during lactation, can affect the babies? So that is a segment 3 study. So that's how. So in male fertility also you do during this, is it affecting spermatogenesis? Everything you do. So that's how you go for a special toxicity study or dermal, ocular, rectal, because many pediatric population you have to give it a rectal formulation. Next slide. So in acute toxicity study, what you do? Quickly go. You just give the dose, okay, in 5 mmol, just repeatedly, and you selected 3 to 4 dose, because when you do in vitro study, you extrapolate the dose for in vivo, and that is considered to be median dose. So you give a lower dose and higher dose. So then you give a single bolus dose and you evaluate all the mortalities. So normally when you plan for any clinical study, we always go for a 27, clinical sign to find out. Like suppose a drug is toxic, how do you get to know? Animal is not going to have food, normally food, or taking a water. You have a loss of fur. So all these 27 clinical signs is been going to evaluate. Okay? And that's the indirect, you get to know that this drug is toxic. Then you go for next repeated toxicity. So from acute toxicity study, what you get? The drug which is applicable, NOEL. What level it is no adverse effect and what level there is a lower. Next. So this is going to inform that, how it is going to help it. And that's how, next, that you get to know that what is the most susceptible, species. You do it in a mice, you do it in a rat. You get to know that what is the most susceptible species. And that's how you go for what dose is applicable, what dose could be overdose. Okay, I said that during this time only you decided that if it is overdose, how the mechanism for a physician to manage in case of a poisoning with this drug. Okay, next slide. So if you look at this slide, you can see that if you work on a couple of molecules, you get to know that which one is less toxic compared to B. But if you get it like D and C, if though C is looking like less toxic, but usually when you clearly see that it is compared to the D, it is more toxic. And that's how you have to have an interpretation. Next slide. So then you go for a repeated dose. Let's go quickly. In repeated dose, you similarly give the dose and see that how the drug is going to behave. And you also confirm whatever dose you are getting, NOEL and LOEL, in repeated dose. So you give the drug for 28 days, 90 days, 6 months, 1 year. They all depend on clinical use. Next slide. So then what you do is what you observe, whatever observation you get it in clinical acute toxicity study. Next slide, please. So you go for all the extract, like how about hematology, effect on blood, effect on tissue, and all are confirmed by histopathology. Go to next slide. Okay, this is working. Fine. So as I said, it is an important part. You know that there are a lot of inspection was done by USFDA during the COVID. You know the reason? Reason is all regulators, they have their own database. Many of the innovator company they put as non-mutagenic or mutagenic, but another company put it as a mutagenic. So this is very important that how do you establish the drug can cause mutagenicity or carcinogenicity. So this is how in vitro test is done. Ultimately, once you do that, any drug used for more than 6 months, you have to establish can it cause cancer, any chronic disease. Okay, so you do give the drug for more than 2 years, you find out that can it cause carcinogenicity. So this is how it is done, that in rat, more 50 per group, and you also use a positive marker. So that is why. So as I said that many of the drug has been withdrawn from the market because it causes cardiac arrhythmia. So nowadays, any drug you're going to approve, you have to see that can it affect other systems. Happen to go to safety pharmacology, society, they'll say that they will deliberate, they'll put the academic people, regulator, other industry people. So any guideline they come up, they always say that whether how to implement it. So you have studies that can it affect reproductive, can it affect, respiratory or CNS. Every studies you are going to do it. Now you might have seen that. Do you know any drug causes SLE-like syndrome? You got a point. Many drug causes autoimmunity. So what you do during the initial phase, you can just see that even vaccine, any biosimilar, you need to find that how many of them can develop autoimmunity. So that is why immunologically toxicity, you do it during this phase, and that is why. So meantime, if you go for all regulator, any clinical use, you are going to do 3 times more than preclinical studies. And that is what the concept is, that any clinical use, you're going to more than 3 times. So this is what I talking about pregnancy. Can the drug is safe in pregnancy? I'll give you one example, simple example. You know that we do not recommend aspirin in pregnancy, but otherwise it is safe. Why? Because if the pregnant woman take aspirin and all NSAID, non-steroidal anti-inflammatory drug, they have a vasoconstrictor properties. Okay? So if they consume, then placental circulation will be affected. So what will be result? Result will be low birth weight baby. Okay, so that is why many drugs, though it will not affect directly to pregnancy, but it will affect the babies you are on. So you have to see that, can it affect the fertility, like implantation? Can it affect the teratogenicity or prenatal? When you give the drug during the lactation period, is it going to affect the babies? And that is why you do all the studies during the time. And similarly for male fertility studies also, is it going to affect the male fertility? You are going to do it. And that is why you do all male fertility and female fertility tests. So that's how you go for it. Now, what about dermal toxicity study? Any topical application, what are the practical point you are going to do it? Is the drug is irritant to the skin. Okay? So if it is irritant, then you get the result is redness or itching. So this all done in an animal which is called guinea pig or rabbit. Especially people the guinea pig, because guinea pig is 1,000 times more sensitive to histamine. So this is how you do for a guinea pig or vaginal toxicity study, then rectal tolerance. Why do you do rectal tolerance? Because many pediatric population you cannot give it orally or IV. So you have to give a provision. Can, if it is required in infant or a children, so that is why they do rectal tolerance test is there. So any parenteral drug, when you give it IV or intramuscular, what data is required? Is it suitable to give? Because the reason behind in vaccine, you might have seen that how many of them they have a nodules or a itching or a pain at the side of. So when you give it to the animal, what you do is you take out the skin, you go for a histopathology. All this data is initially generated, and similarly for ocular toxicity study. So in ocular toxicity study, what you do is, you give this drug in a topical preparation, and you do ocular kinetics. If you remember the clock, you take out the sample at 9 o'clock, 3 o'clock, 6 o'clock. So you establish ocular kinetic and how much it goes to systemic circulation. So that's how you do ocular inhalation toxicity study, allergogenicity, lymph node. Many drug causes pseudolymphoma. One of the example you can take is a phenytoin. So during that time, like you say, can it affect the immunological mechanism? Can it affect the lymph node? So initially you do the study, then all the dynamics, pharmacokinetics. So now we call it PB, PK, PD. What is a dose? What is a dynamic effect? This is PK/PD. Okay. Now it is called physiological based PK/PD. So basically, this is a mathematical model. So preclinical development, you establish how it is going to develop, effect. So this is how P2 prolongation is done. So ultimately, what you do, any drug you select, you identify lead or hit or lead compound. So you are going to do, is it effective? Is it safe? How about new chemical entity? Do you know how it is absorbed, how it is metabolized, how it is excreted? So once you do that, you basically see the goal for non-clinical evaluation is characterization of toxic effect or target organ. You know that every drug, they have a positive effect and negative effect. So what you have to do is how much you can extract the maximum benefit. Can you minimize the adverse effect? Like if you take an example, again, I'll give it paracetamol. Everybody knows, if somebody asks it, most commonest poisoning for hepatotoxicity is paracetamol. But still you use it because you know the dose, okay? And how do you manage it? So ultimately, you need to know that dose dependence, relationship of exposure, preclinical reversibility. So ultimately, you need to decide what dose will be effective. in human. So that is why you do phase 1 study, and ultimately this is a guidelines for doing a preclinical study, how do you go for a clinical studies. And also you can say that, like, you know, all these forms, like you say, you happen to go to US FDA, you fill 1572 form, and it all talk about requirement for applying to phase 1 study and subsequently NDA application. So these are important, and animal toxicity studies, this is manufacturing information. So ultimately when you apply to IND application, so you are going to submit with a phase 1 protocol, and that's how you do it. Next slide, please. Okay. I just wanted to summarize. When you make an application as IND application, what you get, that all the people, medical people, chemistry people, pharmacology, toxicology, statistical, they are going to evaluate. And what is the time period for US FDA or Indian regulator? It's 30 days. So within 30 days, we have to evaluate all application, and ultimately, you know, you are going to get an approval, go and no go. So go means you're going to go for a phase 1 study. But please remember that we have two experience of phase 1 study. Earlier phase 1 study, we are going to recruit all 12 to 24 volunteers. But there is an incident took place in the UK. There is a biosimilar, one of the US company, they hired a CRO in Europe and they did a study in UK. And what they did is, this biosimilar was injected to 12 volunteers. And all 12 of them, they developed a gangrene. Okay. Second incident took place in France. There a big group of cannabis, they were working on it. And they extrapolated dose for a human, and some of the volunteers, they died. So whole France regulatory under the scrutiny because they could not find out the appropriate dose to be selected for healthy volunteers. So nowadays they say that, you know, if you plan for a phase 1 study, just complete 1 volunteers, participant. If it is safe, go to the next one. So in order to have a safety margin that is it safe, go 1 by 1, and that is what the provision is. Okay. So as you can see that by the time we have been doing, we are learning some experience, and based on the experience, we are going to do it. Okay. Now, recent concept is coming is healthy volunteer studies, healthy human volunteers. It was actually started in Oxford. We are also coming up with, you know, guideline for human volunteers. You need not have to go to preclinical like vaccine studies, malaria studies. We can go for a human volunteers. So this has been also new concept. So it is going to do it. So these are some gazette notifications for new drug. Go to the next. So any study you do, you must register in CTRI. You must register in clinical. Every country, like Malaysia, have a registration. Every country have a registration. So basically, you know that any study you do, you need to have a transparency. Today, you wanted to see that how many COVID trial has completed. Just go to clinicaltrial.com. Just go to any of the registry. And second important point is, if you do not register for any clinical studies, nobody will going to accept your paper. I'm a chief editor for 3 journals. I'm not going to accept this paper if you do not register initially. Okay. So once you register, you get a registration number, and this number is very important when you send for a publication, and that is what transparency is there. So there are some recent development in India, but any drug you develop, definitely you have to go for a test license because you only allow to do clinical study if the drug is PPRD GMP condition, good manufacturing condition. Okay, and that you call it a current GMP. So this is what you require, that current GMP, but we have a waiver procedure also. That if a countries have a prevalent for, like we extended, like we have approved Bidacoline as an extended phase 2 program. It was not completed phase 3 study. So those diseases we are prevalent, like tuberculosis, malaria. So we can go for a waiver if somebody has done a study in outside. And we have a subgroup population of South Asian or Asian population, you can go with approval. So this is what public relations. So we have a national health policy. Every country has a national health policy. They look at the ethical standard. They look at the regulatory standard, and that's how we give an approval. So this is what we did it in Corona. That how many drug has been approved. So this is what we learned, that when we talked about Mars, that everything was closed. We do not know how it is going to reopen. Of course, now it is reopened. But during this time, you might have seen that a lot of drug has been approved as emergency approval. Okay, so this is what emergency approval also, you have a policy to generate the data of existing data, and can you give an approval for existing data? So this is what that, when you have a challenge, we learn an opportunity, and that is what our leader says. So with this, I thank you very much. Next. Jai Hind. So you have any questions, I'll be happy to answer. If you have time. Any questions you have? It's an experience of looking for a new drug. Okay. So if you talk today that what are the leading countries they're looking for a new drug, it's all multinational companies, 90%. Of course, academic institutes are also looking for that. They outsource the patent to the multinational company for doing a clinical trial. So in India, we get at least 3 to 4 applications in a month. So that means if you look at 70s, 80s, now every month we get a new drug. And as you know that, we are very good at formulation, new formulation in India. So that is what we have an experience. Any questions?
Good morning, everyone. Thank you so much, sir, for the nice presentation. So my question is, as you just the new entities are coming up, the new drugs are coming up. So it's taking a lot of time for preclinical trials, then the clinical trials, then the post-marketing surveillance, and then it is approved. So what strategies we can follow to reduce the time consumption, and we still are utilizing a lot of money on these things. So what process or things we can follow to minimize the time consumption and the amount of money invested in this? Thank you.
Okay. So you might have seen that earlier used to take 20 years time. Then we implement, hypothescaling and other hypothescaling to screen the molecule in a short period of time. So after that, there's a concept coming is microdosing. Microdosing means you do not do preclinical study. You just give a small amount of the drug into the human and you establish a PKPD modeling in order to shorten the time. Recently, USFDA had implement a concept note that you may not have to go for a preclinical study. You do all cell-based study. Recently, in last month, Indian regulator also has approved cell-based studies. So these all that if the drug is safe and effective, you know that you can do the cell-based study. You need not have to go for a preclinical study. That is what the concept is going on to reduce the time. Okay. And also for a clinical study, there is accelerating clinical study, accelerating phase of clinical study. So during COVID time, you might have seen that there are a lot of change of concept. We call it decentralized clinical trial. Have you heard that word? Decentralized. So that means patient will not come to the hospital, he will go to the house. Okay, adaptive design, CT1. There are a lot of change of concept in order to reduce the time so that the molecule can immediately come in a short period of time to the clinics and patient will get a benefit. But regarding phase 4 studies, it has been strictly implemented. If you approve a drug as a waiver, but you have to have a sound mechanism of phase 4 study, PMS studies. That need to be done that because we will not get what is a real world scenario once a drug is approved. So that has been implemented. If you go for a waiver also, you have to implement that phase 4. Thank you.
One more question, sir. As you give the example of the drugs and their toxicity effects, so there are companies they are hiding the undesirable effects. So by what ways we can come to know about so we can minimize those side effects and all?
Okay, so we have a mechanism. Those preclinical studies, it is done under OECD. So under OECD, there is a quality assurance team. They go and look at the data. And from GLP monitoring authority also, we go and look at the data. Basically, when the inspector goes, they look at the data. Can I reconstruct the whole study? Similarly, for any clinical studies, we have an inspection under ICS. So there is a monitor which is appointed by a sponsor who look at the data, and from drug inspector also the lead team it goes and they look at that all raw data, means source data, and they look at that whatever they submitted, is it authenticated? They verify. So every audit and inspection is done for each and every data, for raw data and source data.
Thank you so
And
much.
this data is kept it for preclinical study, we keep it for 9 years, 3 cycle. For clinical study, we keep it for 5 years. So during that time, anybody, but it's up to the regulator. If you happen to go to Brazil, they keep it for 15 years. So that is a problem.
Thank you so much, sir.
Thank you. Any other question? Okay, thank you very much.
Okay, thank you so much. First to the chairperson, Associate Professor Dr. Masila Mani, for introducing the speaker. And thank you so much for the speaker, Professor Dr. Vikash Mehdi. It was a wonderful speech, very eye-opening. All right, now we shall be having a short coffee break, which will be until 10:45. Please come back by 10:45 because we have to finish the oral presentations in time for the Friday prayers. With that, thank you. The coffee break would be at the spine level 2. So we have to order in. by our Doctors Sabah Muhammad Abdul Al.
Assalamu alaikum warahmatullahi wabarakatuh. Good morning, respected guests, distinguished speakers and presenters. A very warm welcome to MAHSA International Medical Technology Conference 2023. So, before we start oral presentation, I would like to extend our thanks and warm welcome to our judge panel, Prof. Dr. Bikash and Prof. Dr. Subhash. Before starting the oral presentation, I would like also to brief regarding the presentation process. So every presenter will have 10 minutes for presentation, followed by 5 minutes to answer the question of the judge. And to ensure that the presentation process is smooth and fair, there will be 3 times frame kill. The first one will indicate that the presenter will have 3 minutes left, and the other touring will be at the 10-minute mark, so indicate that the time of presentation is over and the presenter kindly to conclude his presentation. So I would like to invite the first presenter, Dr. Aguilardo Pilaga. The title of his presentation, In vitro antimicrobial and cytotoxic activities of leaf extract of Sensipitia tripiciata against colorectal cancer cell lines. Okay, doctor. Okay.
Good morning, all. I am Dr. Kalaraju. I am senior lecturer in MAHSA University. I am from Faculty of Pharmacy, Medicinal Chemistry Department. So my presentation topic is today phytochemical investigation and evaluation of cytotoxic activity of Sensiveria trifasciata against various cancer cell lines. So this is the plant Sensiveria trifasciata. So it is widely grown in Malaysia. It is also called as mother-in-law tongue. So different things like it looks like snake. It is a leaves, the Sensiveria trifasciata. It has many traditional uses. Mainly like treatment of many diseases like anti-inflammatory, antipyretic, anti-diabetic. So traditionally many uses are there. So here I selected to study for the anti-cancer study. All of you know that cancer is the dreadful disease. So it is a second leading cause of death globally, and around 19.3 million new cancer cases are coming every year. And another thing is, all of we know that when we use the synthetic drugs, there are many toxic effects, long-term use. So it is better to choose the natural therapy for cure. And the species belonging to the plant belong to Agaveaceae family. So it possesses, as I told, it is used for 3 different diseases. So the plant possesses many bioactive phytoconstituents like steroids, glycosides, flavonoids, saponins, alkaloids, terpenoids, tocopherols, and tannins. So, and till now, there is no report about the anti-cancer studies with mainly on this plant. So, briefly about the literature review, as I told, it is used to treat many diseases, to treat the boils, fever, and swelling. Okay, so the pain, the relief pain, hemorrhoids, and also management of asthma, malaria, alopecia, and it is used as antidote for snake poisoning because of presence of tannins in the plant. Also, different other uses. So, like antioxidants, analgesic, antipyretic, antimicrobial. So, other uses are there. Antidiabetic, thrombolytic activity. So as I told, the plant contains mainly the different active constituents, saponids. So it consists of mainly the phytopenoid saponids and also flavonoids. It consists of different flavonoids containing the flavone ring, flavonols, and also contains alkaloids and the terpenoids. So mainly I want to do the hypothesis is the leaf extracts of Sansevieria trifasciata probably possess promising anti-cancer activity against various cancer cell lines. And to check the apoptosis. This is my main hypothesis. So significance of this study, so mainly to explore the anti-cancer activity of Sansevieria trifasciata leaf extracts against mainly the pancreatic cancer cell lines. So ASPC1 lung cancer cell lines, A549 esophageal squamous cancer cell lines, KYSC150, and rhabdomyosarcoma cells, RD cells. So these cell lines I selected for the study, and also the Vero cells for comparison. So and also some of the main objectives is for the phytochemical screening of the plant and to evaluate the anti-cancer activity, and also the last main objective is to evaluate the apoptosis induction activity of the selected extracts by using the capsaicin 3/7 assay. Capsaicin. So as usual, we have done actually this is final year project, students and all together we did. Okay. So we took the plant as usual. We extracted the plant by using the different solvents according to the polarity. So we selected the solvents hexane, ethyl acetate, and methanol. So we extracted by using the Soxhlet extraction method. And we did the MTT assay to find out the cytotoxic activity. So in this process, we did first the cell seeding. So cells, the grown anti-cancer cells, cell seeding, we did. So cells were plated in 96 well plate. So with the density of the mainly the cells of 4 cells per well in 100 microliter cell suspension solution. Then cell treatment, the confluents are treated with the extracts with different doses. So the doses is selected is starting from 1000 microliter. So serial dilution 1000 microliter, 500 microliter per ml, and 250 microliter, 125 microliter, 65 microliter, 32 microliter, and last is 15 microliters. So the extracts were treated, the cells were treated with the extracts and incubated for 24 hours. Then the MTT assay, we added the MTT reagent, 20 microliter, and again incubated for 4 hours. Then we added the 100 microliters of sodium surfactant, sodium dodecyl sulfate, so to solubilize the dye. And so after incubation, then we took the readings, the optical density at the wavelength 5 centimeters. Okay, so this is, and also in the similar way, capsaicin assay method also
we followed the same method. And the data is analyzed by ANOVA method and T-square method, T-test method. So these are the results of the extracts. And we did the phytochemical screening. So phytochemical screening showed that the extract contained flavonoids, alkaloids, phenolic compounds, glycosides, and saponins. So by doing the qualitative tests. And this is the result for the MTTSA. So interestingly, it showed that mainly for the RD cell lines and KYSC 150 cell lines, it showed good cytotoxic activity. Generally, according to the guidelines, IC50 values, IC50 value of less than 30 micrograms, microgram per ml is the good cytotoxic activity. The two drugs have good cytotoxic activity. So we got for the ethyl acetate extract around 30 microgram per ml, and for mainly the RD cells and KYSC cells, 13 for hexane extract, and the value of ethyl acetate 20 and methanolic 14.8. And the lung cancer cell lines and pancreatic cell lines did not show good cytotoxic activity. So overall, the result. So the KYSC 150 cell were showing more sensitive effect on hexane extract at a dose of 13.8 microgram per ml, while hexane extract exhibited weak on the other cell lines. And ethyl acetate extract possesses also very good cytotoxic activity on the KYSC cells and RD cancer cell lines. So this is just the graphical representation of the statistics. So MTT for RD cells. Statistics. And also I calculated the selective index of the cells mainly. So selective index also showed that mainly for the KYSC cells and the RD cells have good cytotoxic activity, because if the selective index is more than 1, then the plant have good cytotoxic activity. It can be calculated by IC50 value. So overall, the cytotoxic activity of Sensoria fasciata leaf extract is measured in the cancer cell lines and the normal cell lines for comparison, the activity. Mainly the ethyl acetate extract and the methanolic extract have good anti-cancer activity. Therefore, the RD cells, the myosarcoma cells and the KYSC cells were selected for further confirmation for apoptosis by Capsa-C assay method. So this is the result. So they showed that 15.625 microgram per ml has good cytotoxic activity. So same result. So these are the morphology of cell lines. So actually we check the morphology of cell lines. So the normal cell lines were clear, and the cytotoxic cell lines, they shrink, and there was changes in the shrinking of the cells. Mainly the cytotoxic activity is mainly due to the presence of secondary metabolites. So
like steroidal saponins are the main responsible for the cytotoxic activity. So overall ethyl acetate extract, as I told, is showing good activity on RT cells and mainly the KVSC cells.
Okay, thank you, Dr. Abilario, because we have to fix the time.
The mainly conclusion. So, yeah. So, thank you.
Okay, thank you. So now I would like to invite the judging panel to provide the question session.
Yeah. Did you identify any active ingredient? And whatever dose you have found cytotoxicity effect, did you also correlate with, what will be the tolerable dose? I'll just repeat the question. Did you identify any active ingredient? And the cell line you find cytotoxic effect, did you correlate with maximum tolerable dose in terms of, what will be acceptable one?
So, mainly, according to the guidelines, means less than 30 microgram per ml is the effective dose. So we selected below that dose for the study.
Regarding any comparison with some past drug, like doxorubicin, it is on the top of
Yeah.
any comparison.
We did compare only with the normal vero cells only. We compared.
Now, in any studies, when you go for a new chemical entity, it has to be compared with like a positive control. What is the standard therapy? Is it better than standard therapy?
We are planning to study with the cisplatin also for the study and correlate with the standard therapy.
But okay, yeah. From our study, you combine with the Vero cells, normal cell, and the cancer cell. Which one give very significant differences? You did a 3 extract.
Yeah, 3 extract. Ethyl acetate extract on mainly the RD. It showed
specifically compared with the Vero cells.
Yeah, Vero cells. Ethyl acetate extract and the methanolic extract showed the good significance.
Yeah, next time please include the positive.
Okay.
Okay, so the second or next presenter, Lord Faxi, the title of the presentation.
Ditarik yang tu.
Good morning, distinguished judges, ladies and gentlemen. The topic of my presentation today is photobiomodulation in pediatric chemotherapy-induced leukocytis. Next slide, please. Cancer is a major public health problem worldwide and the leading cause of death. The incidence of pediatric cancer has been steadily increasing over the past decades. And you can see that the most reported ones are the pediatric cancer. In Malaysia, the incidence of pediatric cancer is said to be around 77 million. Thanks to the various advancements in the treatment modalities of surgery, chemotherapy, radiation therapy, hematopoietic cell transplantation, and targeted and immunologically based agents, the death rates have decreased to a large extent, according to the report of 2022. But these advancement has come with a basket full of side effects. Oral mucositis is one of the most debilitating side effects of chemotherapy, affecting around 52 to 80% of the children. This can cause severe discomfort, affecting their eating habits and creating a nutritional challenge and compromising the child's well-being. These ulcerative lesions can act as a portal entry of microorganisms, increasing the incidence of bacteremia and sepsis. Severity of the condition, the unplanned breaks and infection delays the treatment of subsequent chemotherapeutic cycle, and it can overall affect the prognosis of the disease state. Coming to the management of mucositis. Yes, systemic and topical agents have been tried to decrease the severity of mucositis. But today, although few of them have shown positive results, none of them has emerged as a gold standard. This is where the low-level laser therapy comes into picture. Low-level laser therapy is a procedure where a high-density monochromatic light is applied to the tissues. It is applied in a very low non-thermal doses, which is said to be around 0.5 or less than 0.5 watts. Hence, it is also called as whole laser therapy. It is also called as photobiomodulation therapy. This has been postulated to decrease the severity of mucositis via the anti-inflammatory action, increased healing potential, and also having a good pain relief. But it has not been considered as a conclusive tool due to the heterogeneity in the parameters. It has not been considered as a conclusive tool due to the heterogeneity in the parameters that were used in laser, the protocols, and also the assessment tools of mucositis. Hence, we decided to conduct this study to evaluate the effect of low-level laser therapy or photobiomodulation protocol in pediatric chemotherapy-induced mucositis. This was an interventional part of the clinical trial, which was done on low-level laser therapy and mucositis by the Cancer Institute of Kasturba Medical College, Manipal, India. The study comprised of 12 patients from the pediatric oncology ward with grade 2 and more than grade 2 mucositis according to the WHO toxicity scoring criteria. The mean age of the patients were 9 years. Out of the 12 patients, 10 patients underwent laser treatment with oral hygiene instructions and monitoring. The 2 patients who refused the laser treatment on the day of the procedure underwent only oral hygiene instructions and monitoring. The laser parameters were as follows. We used a diode laser of 980 nanometer wavelength with 0.2 watts in a continuous mode and a focal spot of 1 centimeter square. The power radiance and time was calculated for individual patients based on the severity toxicity scoring criteria, and it ranged from 4 to 16 joules per centimeter square with 15 to 60 seconds. This was repeated 3 times for 7 consecutive days. The pain score and the oral toxicity score was recorded at the baseline, 3rd day, 5th day, 7th day, and 15th day by a trained personnel who was blinded to the treatment. Pain score was evaluated using the visual analog scale, which ranged from no hurt to hurt the worst, and the patients who were unable to give a response were excluded from the study. The oral toxicity score was graded based on the WHO scoring criteria with a grade 1 to 4. The results of our study showed that majority of the patients belonged to grade 3 with a pain score of 8, and the severity of incident decreased by 5th day in our study group compared to the observation group. Coming to the discussion, low-level laser therapy was well tolerated by all the patients in our study, And this was in agreement with a study done in the year 2019, which stated that low-level laser therapy could be used as a treatment for oral mucositis. The study done in 2022 stated that low-level laser therapy appears to be a safe therapy, but further evidence is needed to assess its efficacy as a preventive tool or as a treatment tool for oral mucositis in children, because most of the reported cases were done in adult population. Now comes the significance of our study. Our study, which was done in pediatric chemotherapy-induced mucositis, showed that low-grade mucositis required only low energy, and a grade of 2, 3, and 4, a dosage of 4 joules, 8 joules, and 16 joules showed a curative effect. This was in agreement with a study done in the year 2012, which stated that more than 4 joules can give a curative effect. So coming to the conclusion, based on the data results of our study, we can say that photobiomodulation can be considered as one of the preferred options in managing pediatric chemotherapy-induced mucositis if delivered in optimal doses. Thank you.
Okay, thank you so much. And now we will start the question and answer session. So I will give you
Very good studies. I have a couple of queries actually. How the informed consent was taken, and second point is when you look at the methodology. So was it put as an observational blind because you have used the visual analog scale, both the scale were used. So first question is, how did you take an informed consent? Second point is whether you have, implement observational blind.
The first thing, informed consent, the children, as you know, it was below the age of 9 years. So we took the consent from the parents. And of course, we also had to get an agreement from the child. The child should have been willing to participate. And that is why the 2 patients who backed out in the study, although the parents had given a consent, the children, it was not very, they were not very cooperative.
Why am I asking the question? As per our guideline, ICMA guideline for biomedical research, if it is less than 7 years, obviously you are going for parents, whether single or both the parents, if
Yes.
it is very desperate. If it's more than 7 years, then you are going for a viable consent till 12 years. And there is a procedure for written consent also. Second point is since they are the vulnerable group, children are vulnerable group. So did you make it a point of AB recording, audio visual? So as our provision since 2015, if it is a vulnerable group, then you have to go for audiovisual also.
No, we didn't go for an audiovisual.
So there is a provision for audiovisual if you are going for a new mode of treatment. So that is also there. Second point is, did you put as observational blind in order to avoid the study bias?
Yes, that we have taken care
Thank you. Thank you.
Thank you, madam. So I also have a couple of questions. The first one is you said photobiomodulation. How you define this terminology to your study?
The term photobiomodulation is the use of the light. The photons enter the tissue, and actually what happens is it enters within the tissue into the mitochondria, and it interacts with the cytochrome C complex. And this initiates a lot of cascade of mechanisms like increasing ATPs, nitrous oxide, and it modulates the reactive oxygen species. By all these things, it can result in a better healing.
I think for publication point of view, when you send a paper, it is important to also see that other concomitant medication, especially mucositis, it is going to improve people with vitamin C supplementation, for example. So this has not been counted. If it is given, it should be there that whether it is because of concomitant therapy or whether it is because of laser therapy, other comorbid conditions, these need to be mentioned also. Thank you.
So one thing I would like to add here is, see, one speciality or peculiarity of our group was, I should say whether it is luckily or it was accidental, all the 12 patients, they were undergoing a similar type of chemotherapy, all of them were identified with leukemia. So the dosage was same and the expression was same. So, and all other parameters were.
I got the point because it is a uniformity, it is a homogeneous population,
homogeneous.
but you also mentioned about concomitant therapy, other concomitant.
Okay, so, sure.
You justify this method, low laser treatment is better. What are the current methods? How do you like to justify?
So the current methods actually is basically to control the infection first. Normally what happens is when the patient is coming, such patients with mucositis, when they come to the medical site, everybody concentrates on the diseased aspect. So the oral hygiene is avoided there. But they forget the fact that these ulcerative lesions can act as a portal entry. So at present, the treatments are targeted at reducing the pain and controlling the infection. So controlling the pain and improving the oral, the feeding aspect of it, they give certain mouthwashes with lidocaine. It is something called as, you can call it as, some people call it as magic mouthwash, or it is something to remove that necrotic tissue from them, like baking soda with water. They mix it. It is every day they prepare it and give it to the child. And they have also tried out paliferin, which is a growth factor, which is found to reduce the effect of ulcerations. So it's basically the treatments are targeted towards reducing the pain rather than.
I think the first presentation also commented on study design. So any new modalities is come. So basically for ethical reason, we have to say that make it as add-on therapy. Is it better than standard therapy? That's how we establish. Thank you.
Okay, thank you, bro. Kasih. The next presenter, Ms. Pyongyilin. The title of the presentation, Evaluation of Antioxidant Content and Activity of Roasted Coffee, Arabica and Canifora with different growing methods.
Hi, good morning. Before I would like to thank my supervisor, Dr. Young, for giving me this opportunity to present my research study here today. Before I even started, may I know who in the audience is a coffee lover? And have you ever wondered what's the health benefits of coffee? You may raise up your hand. Thank you. That's quite a lot of y'all. So without further ado, I'll start off my presentation. My topic is the evaluation of antioxidant content and activity of roasted coffee Arabica and Canifora with different brewing methods. Okay, so for the research background, as you all know, there's quite a lot of health benefits from coffee. And there's antioxidants, and one of the bioactive compound flavonoid has different anti-inflammatory property which can help us for improve heart function, reduce anxiety and prevent depression, as well as improve cognitive and reduce the risk of stroke. So based on the studies done by Millett in 2021, the green or roasted Arabica coffee are more significant than the Robusta coffee, known as the Canifora. In the studies in 2022 suggested that green Robusta are more significant than Arabica using the infusion method. So in terms of the total flavonoid content, based on the study done by Uslu shows that Arabica coffee actually has greater total flavonoid content than Robusta, despite of different roasting degree. However, there's limited studies focused on their flavonoid content in both Arabica and Robusta coffee. So the influence of these brewing methods and time on the roasted Arabica coffee, as well as the Robusta coffee, are still understudied. Therefore, it brings me to the research objective, which is to compare the antioxidant activity of the total flavonoid content in the roasted coffee Arabica and Canephora in the influence of various brewing methods and the time. So this will be the methodology by using different types of coffee as well as methods. So the label will be according to here. So as you can see from this diagram here, this is a comparison of the total flavonoid content between coffee Arabica and Robusta by using the infusion method. So the Arabica coffee by using infusion method in 1 minute shows the highest total flavonoid content, whereas the RA1, which is the Robusta with infusion method in 1 minute has the lowest total flavonoid content. By looking at this diagram, in contrary to the previous one, the Robusta coffee by using decoction method actually has, even though it's 1 minute, it shows the highest total flavonoid content here. And as we can see, by overall, all of the Arabica coffee by using this decoction method is actually has significant greater total flavonoid content compared to the Robusta coffee. Next, the DPPH free radical scavenging assay. As you can see, the EC50, the lower the EC50, the greater the scavenging activity, meaning will be greater antioxidant activity of the coffee. So as you can see from here, the Robusta coffee and Arabica coffee with infusion method by using 1 minute actually has the lowest effective concentration 50. And in comparison of the coffee by using the same minute, which is 1 minute, Arabica coffee has the lower effective concentration 50, which shows the greater antioxidant activity. Next, by using the decoction method, Robusta by using 5 minutes, which brew in 5 minutes, will have the lowest effective concentration 50, followed by Arabica coffee brewed in 1 minute. So based on my studies, our findings suggest that coffee Arabica brewed in the infusion method has a higher total flavonoid content than the decoction method. So similarly, in the previous research, the results revealed that the roasted coffee brew prepared with the infusion method actually has a higher flavonoid content. Nonetheless, our findings were slightly contrary to studies done by Core in 2018, which demonstrated the decoction method has a higher total flavonoid content than coffee brewed by infusion method in green coffee. This could be due to the presence of melatonin as an attributable factors. So these studies suggested that the biological activities of these melanoidins and other antioxidants activity in roasted coffee, the presence of this particular compound is a type of antioxidant produced during the coffee roasting process, is believed to be one of the factors for these inconsistent findings. So however, to date, the actual relationship between this compound and flavonoid content is still remain unclear and requires more attention. Moving on, with the infusion method, coffee Arabica actually shows a higher flavonoid content when brewed for 1 minute, then it will lead to a falling trend when it's prolonged brewing time for 3 and 5 minutes. Additionally, the optimum brewing duration was found as 1 minute for coffee Arabica with infusion and 3 minutes for the decoction method. Next, for the optimum brewing time for infusion and decoction for the coffee Robusta will be 1 minute higher in 1 minute than 5 minutes. So the similar results were confirmed by a studies done in 2020, suggested that coffee brewed for 6 minutes was found to have a greater flavonoid content than the coffee brewed for 3 minutes. Flavonoid higher with long. Flavonoid will be higher with a longer extraction time, according to studies. In 2020, the kinetic of flavonoid release from tea leaves into beverage is actually higher as the time prolonged. So similar phenomena was seen in my studies as well. Hence, we can recommend the best brewing time for each coffee types. For the DPPH, the most optimum brewing methods to yield the greatest antioxidant activity here for the Robusta coffee will be the decoction method, and for the Arabica coffee will be using the infusion method. The findings were actually similar to the studies done by Uslu in 2002, which is quite recent, where the infusion method is greater than the decoction method among the coffee Arabica, meaning they just compare among the Arabica with different locations. So for the optimum brewing time for Arabica to maximize the antioxidant activity will be infusion method for 1 minute. With prolonged duration, falling trend will be seen. Similarly for Robusta, for the brewing time, the optimum will be 5 minutes to maximize the antioxidant activity, and it's accordance with a recent studies done in 2023. So therefore, we can deduce that the prolonged brewing time, which exceeds 5 minutes, can actually maximize the antioxidant activity with the decoction method. In conclusion, these will be the key findings of my studies. The optimum brewing methods and time can actually maximize the total flavonoid content as well as the antioxidant activity in both coffee types. So for the infusion method, the Arabica coffee will be better will yield higher total flavonoid content by using infusion method, with the optimum brew time for both coffee will be 1 minute. For the decoction method, the Robusta optimum method will be decoction method, as well as the optimum brew time will be 5 minutes. However, there are still considerable discrepancy in the optimum time for obtaining the maximum antioxidant level, specifically for coffee Robusta. And here's the further works that can be done based on my studies. Thank you for your time.
Okay, thank you. Also now is the time for the question and answer session.
Since you were working on caffeine, do you know which indication clinically use caffeine? Any indication?
For my studies, actually.
No, I'm not. I'm coming to your studies. Just general question.
You mean for indication for caffeine?
Any indication, clinical indication. Caffeine is a respiratory stimulant. So in case of preterm babies having a respiratory problem, this is one indication it is used. Now coming to your studies, so have you established, you have a comparative evaluation of affecting on antioxidant. So is there any particular, amount of caffeine and antioxidant you can dig out from your study data?
No, for my study, I haven't gone into the caffeine studies. Mainly focus on flavonoid for now.
Flavonoid.
Yes, flavonoid.
So any use of flavonoid to use clinically?
Sorry?
Any clinically used flavonoid you can name?
Flavonoid is actually it has huge health benefits in preventing Alzheimer's, as well as preventing heart disease, and also it helps to improve the cognitive and reduce the risk of depression as well. So these are the known clinical health benefits from flavonoids. However, there are limited studies that focus on coffee, the flavonoid found in coffee. That's why I want to carry out these studies to find out, to know that.
So have you sent for publication this paper?
Soon.
No?
Not yet.
Not yet. Thank you.
Thank you.
Okay, thank you for the presentation. Are you a student?
Yeah.
Okay. So, can you recommend only one, Robusta and Arabica? Which one do you recommend? Don't say both.
Arabica.
What is the critical with your method, infusion and decoction? Which one is more critical? I mean, which step, something like during decoction, some amount, some flavonoid is coming out. something.
Um,
Do you think which one is more critical? I think decaxon may be more critical.
critical as
Like
in,
critical in the sense you say coughing is important. It's a compound. Yeah? Another one is flavanol.
yes,
Yeah. So to release this kind of compound, which method or which step might be the critical?
Actually, for both coffees, they have different. By to yield the best total flavonoid content, they actually need different brewing methods. So let's say for Arabica, to yield the greatest total flavonoid from here, we will suggest using infusion method. So the common infusion method for the coffee beverage is such as espresso. So by to maximize the total flavonoid content by consuming it, we recommend taking espresso.
Where you get the sample? The Arabic and Robusta. You buy it from commercial source.
Sorry?
Where you get your sample? Like
I purchased from a local cafe that they import it from.
commercialized.
Yes.
Actually, for studies, when you procure any of this ingredient, it has to be certified. Like you would get it from Sigma or any of this. So that will give it a purity.
Absolutely.
That's all. Yeah.
Thank you.
Thank you.
Okay, thank you.
Thank you.
The next presenter, Ms. Soyi Wai and Mr. Tojarshin. The title of the presentation, the relationship between physical activity and cognitive function among MAHSA University students in health-related health programs.
Okay, good morning everyone. My name is Gulshan and together with me is Sui Wei. We'll be presenting our study, relationship between physical activity and cognitive function among MAHSA students in health-related programs. So I'll go through a brief introduction. Okay, according to WHO, physical inactivity has been a leading cause of mortality, and the numbers are even greater in Malaysia. So, physical activity is a known modulator of cognitive function and has shown a lot of health benefits to us. And according to a local survey led by DC, 43.7% of university students is physically inactive, and factors contributing to this may include time constraints, lack of access to sport facilities, and socioeconomic factors. Continuing, the main objective of our study is to investigate the effect of physical activity on cognitive function among our MAHSA University students. So, the method we use for our study design is the quantitative experimental with one group pre and post test to investigate the effect of increasing PA levels for 2 weeks on CF. Inclusion criteria for our studies will be MAHSA students studying in our programs that include medicine, dentistry, pharmacy, nursing, bioscience, and health science, which are all medicine related field. So participants must be ambulatory and does not have any underlying disease such as cerebral bronchial asthma, uncontrolled seizures, recent surgeries, or even severe musculoskeletal injuries. We obtained written informed consent from all our participants, and our participants are all aged between 18 to 30 years old. We use a formula provided by Daily Brook and Brook to calculate our sample size, which is 173, and we obtain another 10% to compensate for dropouts, and the data we analyze is 168. So for our pilot study, we use only 10% of our main study. To evaluate the physical activity prior to our study, we use IPAQ, which is the International Physical Activity Questionnaire, and it classifies physical activity into three categories, which are mild, moderate, and vigorous. Mild being less than 600 METs, moderate being 600 to 3000 METs, and vigorous being more than 3000, which is metabolic endurance. This is the original questionnaire, and we modified it because we want to assess the physical activity for the last 3 months. Okay. So before we use the questionnaire, we also check the validity and reliability using Pearson correlation coefficient and Cronbach alpha coefficient, and the score of validity and reliability is very high. So to evaluate the cognitive function, we use Ericsson flanker test, which will calculate the flanker effect as the difference in response time between incongruent and congruent trials. So this is how it works. We will provide the participant with 5 letters, including X, C, V, and B. So letter X and C will be responded by clicking key A, and whereas letter V and B will be responded by pressing key L. So the respondents are required to focus on central stimuli and ignore the peripheral stimuli, which are not important. Okay. We use a few statistical tests such as Kolmogorov-Smirnov test, Kruskal-Wallis test, Don-Brofoni-Pusok test, chi-square test, and Pearsall-Collier test in our study. So I'll now pass on to my colleague.
So I'll be discussing about the results first. So before we proceeded with the main study, we conducted a pilot study to assess the feasibility of the research instruments used, the study design, and the data collection procedure. So the pilot study consisted of 17 participants. All are from medicine program. They are all aged from 21 to 25, and most of them are Malays participants. And the results of the pilot study were deemed favorable, so we proceeded with a larger sample size for our main study. So this table demonstrates the socio-demographic characteristics of the 168 participants. And so most of our participants were aged from 21 to 25, and they are mostly Malays, and most of them are from medicine. So this is a continuation of the previous table, and as you can see, most of our participants are categorized as moderate physical activity. So from this table, you can see that in medicine, we have a total of 90 participants from medicine program, and most of them are categorized as moderate physical activity, followed by vigorous and then mild. So focusing on these two diagrams, so these diagrams represent the medians of the flanker. This is the median, this line, as you can see, the medians of the flanker effect of the 3 physical activity categories at baseline and at post-intervention. So as you can see from the first figure, you can obviously tell that mild has the lowest flanker effect. As mentioned by my colleague, having a lower flanker effect implies greater cognitive function. So in this diagram, figure 1A at baseline, mild category participants had the lowest flanker effect, which means they had the best cognitive function. Whereas at post-intervention, it was the vigorous vigorous category having the lowest flanker effect with the best cognitive function. So this is another similar diagram, but in terms of the mean. So focusing only on the blue line, you can see this is for vigorous physical activity category. It started at baseline, it was at 110.39, and then there was a remarkably decrease in flanker effect at close intervention, which became 51.98, insinuating that vigorous physical activity had outperformed the other two physical activity categories. So this is a Pearson correlation scatter plot showing a negative linear relationship for vigorous and moderate at post-intervention. So there were studies that supported our findings, such as this from Davranch, which they studied the effects of acute or vigorous activity on cognitive function, and they supported that performing vigorous exercise improves cognitive process, and that regardless of interference. So our study was able to conclude that increasing physical activity for 2 weeks increased cognitive function in the participants with vigorous physical activity having the highest positive influence on cognitive function and lowest flanker effect lower flanker effect when compared to moderate. We recommend that
we recommend that for further studies that using a longitudinal study with larger sample size and with control group. And because our limitation for our study is that we don't have a control group and there were unequal students from each health-related field program, being medicine being the most. And lastly, the participant was categorized with MVP, which is based on the individual grade. So yeah, that's all. This is our reference.
Okay, thank you. Now is the session for the question and answer.
Thank you very much, and good to see the students are presenting well. Like whenever you plan for any studies, you always should highlight methodology. What is the power of the study? Like you plan to, like you have done the studies, so we need to elaborate on sample size collection. What is the calculation in terms of power of the studies? Second point is, you have used a certain scale. So that also need any scale or you modify. Either you use a standard scale with an approval from those who have, created a scale. And second is if you modify, then you need to, validate the scale before you implement it. So that validation data is required. Okay, so do you have answer to that? Sample size calculation and scale validation. Then third point is, that when you already mentioned, it's good to see that limitation of the study, that control group is not there. But otherwise, that when you are establishing the correlation of physical activity and cognition, you always see the dietary therapy, amount of, kind of, tea, coffee, or any other kind of thing. So that also need to be correlated over a period of time. Thank you.
So, did you find any common physical activities among the members you tested?
Any common activities that they do? Some of them do gym, running, jogging. Mostly is the jogging.
Yeah, I think the physical activity and the food habit are highly connected. So, sir, if you ask me, you're supposed to connect something. Okay.
I think when you plan for a study, this kind of study, you need to also put it as a stratified age group. So you can go for a subgroup analysis in case of a male, in case of a female, or you say 22 certain age. Okay, so you can have a couple of subgroup analysis. Thank you.
Thank you. Okay, the next presenter, Mr. Rodney Silva. The title of the presentation, Effect of Phosphate Solubilizing Bacteria on Growth and Antioxidant Activity of Mulberry Spinach cultivated in different fertilizing conditions.
Good morning to the honorable judges, distinguished chairperson, and accurate timekeeper. My name is Rodney Roland. I'm a final student here at MAHSA in the School of Bioscience for the degree of Biomedical Science. So, I'm presenting on behalf of Ms. Catherine as well as Ms. Abinashori. Towards their study on the effect of phosphate solubilizing bacteria on the growth of antioxidant activity of Malabar spinach. Now, towards it, what is phosphorus has been, as you know, is one of the macronutrients needed for a plant to grow well, as well as it towards the effect of its antioxidant levels. Now, one way that a plant can get this phosphate and utilize it is through phosphate solubilizing bacteria. Now, these are microorganisms which hydrolyze organic and inorganic phosphorus that is found in soil, and its mechanism by also helps in the lowering of soil pH, as well as excretion and mineralization of this phosphorus. Now, towards Malabar spinach, on the other hand, is that it has been shown as a natural source of antioxidants and something that is commonly eaten here in Malaysia. Now, the objective of the study is to evaluate the effect of phosphate solubilization bacteria on the growth and antioxidant activity in Malabar spinach. Now, towards the specific objective of it being through the measurement of mean shoot lengths as well as the number of leaves, towards antioxidant level that is done through is determined by the assay of DPPH radical scavenging activity, as well as to see whether with the addition or the lack of PSB is there an effect towards the plant. Now, the experimental design being on the basis of where A being the environmental control, where there was nothing was added to the control group. B is triple sugar superphosphate. C, rock phosphate. D is tea dust, eggshell, and chicken bones. This is our version of a biofertilizer towards something that can easily be done in the home setting. F again is similar to A, being experimental group. The difference is now with the addition of PSB. And the same can be said for F, G, and H, where it's similar to A to D, but with the addition of PSB. Now, the methodology of it is that PSB was isolated and inoculated on the basis of the rhizosphere and non-rhizosphere of the mother plants that were given from Kajang as well as Kampong Pasir in Semenye. Based on that, then it was inoculated on the strains and was taken the best two strains, of which shown about 200% phosphate solubilization, which is done here, as well as then chlorophyll estimation and antioxidant assay was done in the end once the plant was grown. Towards how the isolation of it was that rhizosporic and non-rhizosporic samples were given. Rhizosporic being soil that or bacteria that is taken specifically from the roots of the plant, or non-rhizosporic bacteria that is taken from surrounding of the plant. So say when you're taking up a plant with its roots, any soil that clings to the roots is considered non-rhizosporic. anything that you have to scrape from the roots is considered raspberry. Then zero dilution, and the screening was done with phosphate solubilizing agar medium, such as Piscoia and Berib. Clear zone was established, and based on that, the strains were taken. This was how it was done towards the study of it and to see which is the best of it. Now, the chlorophyll content estimation was done based upon 0.5 gram of the leaves were grounded up and put with 90% acetone, as well as magnesium chloride, as well as towards it incubated at 5 hours centrifuge, and then based on the supernatant, taken absorbance of UV light. Antioxidant assay is similar to a previous study done. There was discussion on similar to where we have done DPPH ravaging antioxidant assay. Now towards the results with the isolation, the one that we have taken this where we have found 7 of the isolates, and like I said, of the best two, MS3 and MS7 was found to be the best at 200%. As you can I see the combination of it. Towards the effect that we see of the growth of it, where the best was found in the experimental groups, correct? As you see that even when looking at the environmental control, that just with the addition of the phosphate solubilizing bacteria shown a growth, much better thing, much better growth of the plant. The best being in triple superphosphate. Here highlights towards the dosage of consortia, which shows that with p-value of less than 0.05, that triple superphosphate as well as regular phosphate has found to be the best. Towards that, looking at that homemade fertilizer of chicken bone, tea dust, and all that, it was found that with it's similar to its previous studies as well as found to be the best. Towards the pH, as we said, that due to the effect of PSB on the plant being that a lower pH value is able to be obtained, that the best was found with chicken bones. Towards it, the chlorophyll content of it is that again, like the highest is found in rock phosphate. Towards the antioxidant level, which we were studying on towards it, is that found that triple superphosphate as well as rock phosphate or fertilizer with the addition of PSB has found to be effective. In conclusion, PSB can be isolated from rhizosphere and non-rhizosphere soil sample and act as a biofertilizer in the agricultural field. It increases the availability of phosphates to plants, hence it promotes plant growth. It increases plant parameters in Malabar spinach cultivates with PSB proves that the organism is able to enhance the availability of phosphorus in the soil, and antioxidant activities of plants cultivated with PSB is higher than plants in the environmental group. Now, towards the looking where this work can be further pushed is that with the characterization of these MS3 and MS7 that we mentioned, as well as towards total phenolic compound and total flavonoid compound of the Malabar spinach, as well as more thorough soil analysis of it. Thank you.
Thank you. So now also this question and answer session.
Thank you for presentation. And since you put it as a future aspect, any eco effect is there, ecotoxicity or eco-friendly?
I'm sorry?
Do you have any data to show that it is, what about effect on environment? Future.
So towards environmental toxicity, we have not studied yet, but we are hopefully like towards the future works of which students in MAHSA will be able to pursue.
So when you study the environmental effect, what are the parameters you look at? Can you highlight some parameters?
Towards environmental toxicity might be that when looking at, say, the soil levels, right? Well, these are what we commonly are now pushing towards, where biofertilizers and such, that synthetic fertilizers being able to break down so fast and able to leach into the soil, correct? And with soil leaching towards the rivers and all cause the problems within our environment. So the studies of towards the levels of soil, as we said, the further work of soil analysis.
Yeah, so that is from the engineering point of view, from medical student. So you can see that is there any effect on honeybees? Is there any effect on fleas? Like when you talk about environmental toxicities, is there any effect on earthworm? So these are the parameters are done for any of these chemical for environmental toxicity. Thank you.
Okay, thank you for your presentation. What is your positive control? Any kind of fertilizer or something?
So towards it, the control of it, correct?
Kind of a positive control. Which one is positive control?
So each group showed 8 plants. So A1 to A8 shows environmental control.
What did you treat?
Nothing. You were just planted.
Okay, that is a kind of general control. So
Yes.
it's a positive control. You treat with any fertilizer or something.
Towards it, where these are the other fertilizer treated but without TSB, and F towards E shows group E shows where, like I said, without any additional fertilizer but just the addition of phosphate solubilizing bacteria.
Which one is the only fertilizer?
Only fertilizer are B to D.
Okay, that is one. And the other one is you did a rhizosphere and non-rhizosphere. So based on your study, what is the major significant difference between rhizosphere and non-rhizosphere?
Towards rhizosphere, the difference between rhizosphere and non-rhizosphere, like due to the way that it's collected. So things like maybe rhizosphere bacteria would be something more closely related towards the plant, while non-rhizosphere might be towards more general effect on the plant.
So based on the result, how do you correlate?
Based on it, I don't think so. They have studied on it specifically, as we have not identified towards what exactly these phosphate solubilizing bacteria are.
Was it randomized? This was randomized?
I'm sorry, I don't understand what this is.
It means the grouping are randomly allocated. How did you do that?
The grouping of
randomly allocated. How do you allocate the groups?
The groups of this, is it?
Yeah, when you do it, it is randomly allocated.
So taken from this, the seedling were taken from the mother plant as well as the bacteria were taken from this.
So randomization actually the processes come from pedigree, and we use for a clinical trial. Now, thank you.
Thank you.
Thank you. Okay, the next presenter, Albert Jong Chong Hao. The title of the presentation, The Impact of Phosphate Solubilizing Bacteria, Consciential Application on Growth and Bioactivity of White Soil, Mumeridica charantia, Growth in Different Fertilizing Conditions.
Okay, thank you. Good morning, all. My name is Albert. Today, I will introduce you my research topic, which is here is the impact of the phosphate solubilizing bacteria, which is Consortia, and the applications on growth and bioactivity of the wild type bitter gourds, which is Mumeridica charantia, work in different fertilizing conditions. As everyone know, actually, wild type bitter gourd or any general type of bitter gourd that has been well known and very famous for its medicinal properties. It contains a lot of like nutrients and then also vitamins such as One of the example is the vitamin C actually. And then the standard maturation duration is about 60 days until it's from seedling stage until the fruiting stage actually, until we can harvest all the fruits and then as well as the leaves as well. Mostly the wild type bitter gourd, not only the plant is highly nutritious, even including its stems as well as leaves, and also in some study it shows the roots also has some medicinal properties. My current research, the objective would be to isolate the phosphate solubilizing bacteria, which is the rhizosphere bacteria, and then also non-rhizo bacteria from the different source of soil sample, which is from different part of Malaysia. And then the other one would be to assess the efficiency of the phosphate solubilization bacteria, which is PSB, to solubilize different source of phosphate from different source of fertilizers, such as organic fertilizers or even the artificial or synthetic fertilizers. My next objective would be to compare the difference in plant growth parameters, which the vegetable cultivated in different fertilization conditions. Another one would be to compare the difference in antioxidants levels, where the wild-type bitter gourd is grown in various fertilizer conditions, to assess the improvement of the photosynthetical potential of the wild-type bitter gourd via chlorophyll estimations, with and without adding fertilizers. Lastly, this one only has a qualitative test to measure the antimicrobial activity of the plant crude extract, which is the bitter gourd extract. My research design will be follow this flow. Land plot design will be carried out first to allocate the space for the plant growth, and then at the same time I will do a soil collection, which is for the PSB selection, also isolation as well. Once the PSB is selected, which is done by the serial dilution, and then also to isolate them and then to check them on the picoscium media, which is to show the halozones, which later I will show you the example of pictures. Once the PSB has been isolated, I will prepare them in the solution, which is ready to be inoculated on the plants. During this time, and then the fertilizer will be prepared, and then also the plants already planted like on first week. Because the plant from seed until seedling stage, we are not able to inoculate all of these things, the fertilizers into the plant. So I will have to wait for 2 weeks time until the inoculations can be done. Once the fertilizers preparation is done, I will inoculate it with the intervals of 2 weeks. The inoculations will be 3 times, which is from 16 until 60 days. Once the inoculation done, I will measure the plant's parameter, which is the height of the plant as well as the leaf count. Once all these things done, and then the plant's extract will be done on the day 60, which is the maturation of the bitter gourds. And then the plant will be extracted by soxolite extractions. I will extract the plants with soxolite and then with the ethanol as solvent. And then the plants will be tested for antimicrobial. Antimicrobial will be the last one, and then antioxidants, chlorophyll estimation as well. Before all that, in the meantime, the PSB, the consortia, will be done to estimate the efficiency of it to solubilize different source of phosphate. This is the fertilizer treatment that I have. I will give these 4 types of fertilizer to the plants. First one will be the TSP, which is triple superphosphate. 10 grams of TSP was diluted into the tap water only, just one normal tap water. I've used 10 grams because more than 10 grams, which will kill the plants actually, which will become too toxicity for the plants. So I will limit it only to 10 grams. Same goes to urea and potash, as well as vermicompost. And then consortia will be 5 ml per dose added to the experimental group, which is D, E, and F. Here I will show you the result of the efficiency of the isolated PSB in solubilize or to release the phosphate in different source of fertilizers. Here you can see the control, which is only bacteria inside. There is not any phosphate or any other source for them, so it remains at the bottom there. there's no any numbers reading. And then with the second one would be the consortia, which is the doesn't treatment to any of the phosphate as well. And then for the third one, the consortia plus TSG and then TCP, which is the triple superphosphate and then tricalcium phosphate. Here we can see from 0 to 24 hours, there's high numbers of peak here for the release of phosphate, which followedly and then will be slowly reduce the number as the phosphate has been fully utilized by the bacteria. And then the last one here, we can see the bond and TCP. From the 0 hours here, we can see like the phosphate number is quite high and then slowly being utilized. And then why this one quite slow? Because the bacteria slowly digest the bonds to use it as a phosphate. That's why it's slowly increased. This one study can be further increased until more than 96 hours. But due to some limitation, I only proceed this one until 72 hours. And then for this one here will be the plant crude extract study. This one is the plant parameter study. This one is the average height of the wild type bitter gourd from day 16 to day 30 and then day 44. I've grouped the plant, my plant in 6 groups, which is with fertilizer. This one, first 3 will be the environmental control without fertilizer, TSP only, and then vermicompost only. The down here 3 one will be the consortia without fertilizer, with TSP, and then with vermicompost. Here we can see the one with this one with the consortia with TSP were always higher because they have consortia and then they can effectively utilize the phosphate that are treated to the plant, so they can grow up very high at this stage. As for this one here, this one only TSP and it shows like quite slow significance in affecting the plant's growth. And then the most lowest one here, this one is without any fertilizer. It remains at the bottom actually. And then for vermicompost, here is what we're looking for actually, because we're trying to compare the synthetic fertilizer and then also the vermicompost, which is act as the fertilizer for the plant. And it shows not as effective as the TSP introduced to the plant. Here we can see the same patterns also for the numbers of leaves that we have found in the bitter gourds. It shows the exact same patterns, which is the plant's consortia with TSP will be more highest reading. Here we come to the laboratories, the plant crude extract here. The chlorophyll estimation of the wild type bitter gourd, which we can see here, this one. These two will be the TSP group. TSP group shows the highest number here reading because here we can see that actually the PSB, which is the consortia, can greatly improve the chlorophyll reading of the Momotica carentia, which is the bitter gourd. And then for vermicompost, it shows slightly increased actually compared to this one without any fertilizers group. This one here, this one is without consortia, which is no any bacteria introduced to them. This one is with bacteria introduced to them. For this one is the DPPH, which is free radical scavenging activity. Same goes as well. We can see here TSP group will be more higher in reading. And then this one here we can see significance where when the vermicompost was used as fertilizer, it can show increase in reading for the free radical scavenging. And then for here, this one is no fertilizer, so it not that effective. It struggle to survive here actually. And then here in my study, I also studied the total flavonoid content of my wild type bitter gourd. Here I can found that the group with TSP shows like 577. And then this one would be fertilizers with the TSP and then consortia also. Yes, I'll go to this one. This one similar pattern as well. And then this is the TLC, which is to confirm that my plant extract is successfully done. This one is just qualitative test, which shows that the plant crude extract is no effective in inhibits the bacterial growth, but this one can be done in further study. As a conclusion here, I can say that actually the introductions of the prostate solubilizing bacteria can actually greatly improve the antioxidants level of the wild-type zygotes. And then more further study can be done as due to time limitation. I can only done until these three antioxidant level tests. Other tests such as saponins test or anti-diabetic test also can be done in the future. That's all for my topics today. Thank you.
Thank you. Thank you so much.
Good presentation, and I'll just come to the end. Like you showed the last slide that on effect on antibiotic. So basically, we are looking for any compound which will be effective in drug resistance. So did you take a strain, Staphylococcus, which is drug resistance? Or
yeah, actually, this one I took the Staphylococcus. This one I used the control, which is the tetracycline.
Yeah.
And the effective one would be 25 mm, and then here shows 33. But comparison with this one here is 14 mm, so it's like maybe due to.
But when you look at the data, tetracycline is very limited use.
Yes,
so
sir.
you should always go for a standard treatment guideline, which is first choice for a particular indication, second choice, third choice. So that is what he is trying to stress on compared to the positive one, standard. So that's how you go. Thank you.
Thank you. What is the composition of our consortia? You have a group consortia.
Sorry?
You have different treatment, vermiculate, some fertilizer and consortia. Can you open?
This one here.
No, still. Yeah, what is the last one?
This one is actually the experimental group here. Okay. Because I have 6 groups. I have A until A, B, C, D, and then D, E, F.
Okay. Here I understand very compost.
Mm-hmm.
And what is the consortia?
Vermicompost,
Last group. Last group.
This one.
No, I mean, ignore this one. So here.
This last one.
Yeah. Now, what is vermicompost?
What is the meaning of vermicompost? Vermicompost actually is the organic compost which contains of like earthworms, and then it's also including the cow poops as well as goat poops.
Okay, yeah, so it has something, yeah? Because I have some compost, it's a compost. Yes. What about next one?
This one concentrate. Concentrate actually. It actually is the type of applications that I isolated from the prostate solubilization bacteria. I dilute them into water and then extract them, and then we'll make it as a consortia actually. So it's a PSB bacteria inside.
Okay, can you open the TLC?
TLC.
What do you want to say?
Here I want to say is the plant. This one is a plant extract actually, and then TLC here I will show actually the extract was probably extracted from this line here. This one is a thin layer chromatography. This one is a TLC paper. When because this one here actually we can see the line here. This one is the alcohol travel range actually. So as far as this travel from down here to top here, which I can indicate the extract is properly extracted. The plant extract.
I cannot see like a spotted place.
It starts from here actually. It started from here and then go up to here. And then here you can see actually there's 2 colors. This one quite colored. This one a bit like wet.
So it looks like you isolated only one compound.
Yes, sir.
Thank you.
Okay.
Thank you so much. The next presenter, Ms. Tanaja Biramo. The title of the presentation, Knowledge, Skills and Compliance of Nurses on Central Line Associated Bloodstream Infection in the Cardiovascular Department of King Faisal Hospital and Research Center.
The last presentation is not there.
One is absent.
One is absent.
Yes.
So I have
Change the slide.
Number 5 absent.
Okay.
Yes. So now
Number 5.
number 8.
So give me one paper so that
Very good afternoon to the judges and also to the participants. So I'm going to present my topic, knowledge, skill, compliance of nurses related to central line associated bloodstream infection in cardiovascular department, King Faisal Hospital and Research Center, Riyadh.
Pointer.
Oh. So this is a central line catheter. You can see the catheter is inserted into the heart. It's an invasive line, and normally the location of the central line, it could be either internal jugular, external subclavian, or also femoral. So this is the CVC dressing that included in my studies. So this is the one that I'm going to investigate and also to examine my nurses. So this is the flushing and also the clave change. So the introduction, why I chose this topic, because CLABSI is a central line associated bloodstream infection, and also is very critical. And also I believe nurses' knowledge, attitude, and skill are very important in CLABSI prevention. And also particularly nurses in the cardiovascular setting, they have a direct contact, especially in dealing the CVC management. So they should be knowledgeable and compliant when during the insertion and also the management of the central line. So what is actually CLABSI? Is a primary infection of the blood that affects the patient, those who have a CVC line, within 2 calendar days, either with the insertion or removal of the CVC. So majority of the CLABSI are related to the gram-positive organisms such as Coagulase negative Staphylococcus and also gram-negative organism. So the problem statement, what is the primary concern that need me to do this research? This was my master's thesis during when I was working in Riyadh, Saudi Arabia. So I worked in the cardiac ICU. So the concern was there was a lot of inadequate maintenance and care of the central line that contribute to CLABSI. And also we use the National Database Nursing Quality Indicator, which we compare with another country, which is United States, 500 bedded with 500 bedded, I think minimum. So based on that, we do the quality indicator interpretation of CLABSI. So the cardiology department, even though the cases of CLABSI was very high in 2017, and also the number, even though it's decreasing, but actually we are aiming at a zero harm hospital. So actually we want to have a zero CLABSI. So my study aim is to assess the nurses' knowledge, skill, and compliance with the central venous catheter management. So these are my research questions. Is there any association between knowledge, skill, and compliance with the CLABSI? Is there any difference between the nurses' knowledge of the CLABSI pre and post-test? Can there be any association between nurses' experience with post-test and pre-test knowledge? Is there any association between nurses' level of education pre and post-test compliance and also nurses' experience with their competency skill? So this is my research hypothesis. So my hypothesis was knowledge of the pre-test score will be lower than the post-test. And also null hypothesis is knowledge pre-test score will be same as the post-test. Experienced nurses will have higher score in the pre and post, and also null hypothesis is the nurses will have the same score in the knowledge pre and post-test. So this is the conceptual framework the one I formulated for my research. And these are the literature reviews done that are mainly like focusing on the importance of the educational and also the training for the nurses in order to prevent the CLABSI. And this, the setting of the study was done in Riyadh. It's a tertiary hospital. It has like 1,000 physicians, and we have 67 nationalities working in that hospital. And mainly I conducted this study in the cardiovascular department because most of the patients are mainly post-cardiac surgical, medical, and also cardiology, and they have central line and also PICC line. So material and method, actually we conducted a prospective interventional study. Mostly 180 to 200 nurses are working in that in 5 departments. Convenient sampling was used because it was easy, and also I don't have much time to do a random sampling. So 80 nurses were selected after I obtained the consent from the participant. And what was my inclusion criteria? They should be registered with the Saudi Council. They work in the cardiovascular department at least more than a month. They should be fully employed. Exclusion criteria was the float nurses, intern nurses, care assistant, and those nurses less than 1 month of experience. This is my sample size calculator where I used to recruit 80 participants, mostly are the nurses. So ethical approval was obtained prior to this conducting the study. And also I get the consent also from the participants, and a short briefing was given to them how to answer the questions and all those things. So what are the instruments that are used? There is a 3-part. First part is the demographic data, which included age, gender, education, years of experience, whether they are single skill or double skill. That means whether they have pediatric or adult skill. And what are the shift coverage? Either they are doing day shift or night shift. The second part, I use the evidence-based policy and procedure from the King Faisal and also CDC guideline. They will ask, the question were yes or do not know. And we will enter the correct answers. If yes, we concluded as 1 and do not know as 0. So these are the things. So I have knowledge related question which was based on the policy, compliance related question based on the CDC bundle, central line care, and also the chlorhexidine bath, and also the competency based skill where was the skill check. I used the CVC management, needleless adapter change, and also CVC dressing plus the tubing change. So this is the demographic data. You can see that mainly, I think Asian nurses were the highest population and female was the highest. This is the knowledge question, and these were the compliance questions. And the third part, I used these are the competency based from King Faisal, which was approved by the hospital. So the CVC management, the flushing, and the dressings. Validity and reliability of my questionnaire, I use, even though I took it from the King Faisal, but I use the clinical instructor, case managers, everything to review the questionnaires, and we pilot before we give it to the nurses. And the data collection procedure, I handed over to the nurse manager and the clinical instructor. The participants were given 10 minutes to answer the pre-test. And then after that, we do the education. We did the education for them to, practice. And also after a month and 2 months, we did a post-test. The same participant will do the post-test. So we did the pre and post-test. Actually, we for the demographic, we use the mean media, and also for the pre and post-test, we use the mean media comparison. And for the correlation, we use the chi-square. And we can see here, like what I mentioned, Asian was the highest. And majority nurses, they are working both shift. And this is the correlation to say that actually mainly those nurses who have 6 to 15 years, they are more experienced in managing the central line in comparable to those who have less experience. And also you can see the post-test related to Club C and also the compliance question. Post-test, the score is much better, even though not much difference. And also you can see the comparable correlation between the knowledge related to pre and post-test. The nurses with the 6 to 10 years, they have a better score. That means the question is 10, but if they score more than 7, consider their scoring is much better. And this is the correlation between the pre-test and post-test with the compliance. Also same, similar 6 to 10 years, they have a better score in terms of experience. And level of education, we can see the bachelor degree nurses, they have a better understanding and better compliance with the Club C knowledge questions and also the practice. And also this is the Club C, also similar, the same, the level of education, the bachelor degree are having a better. And in terms of the experience, that means the skill check, there is, the 6 to 15 years, actually they are doing better. Those who are more than 15 years can feel their compliance is like much deteriorated. And this is the CLAPSI quality indicator, which we do monthly basis. And I did the study somewhere between July to September. Actually, it doesn't make much difference, but there is some difference because during that period, there was some education. So this is the limitation of my study. The sample was too small. If I get a better sample, maybe I can have a better analysis. And my recommendation, I should have a continuation of the studies, and also they should have more skills and compliance related, because more practice, more skill, it is better to reduce the CLAPSI. My conclusion is good CLAPSI knowledge and compliance among nurses, and also based on the level of education and also their experiences, so they can do better. And these are my references. This is the ethical approval from Research King Faisal. This is a scientific committee approval. And these are my questions. And these are the participant consent, where I need to translate between English and Arabic before giving it to the participants. Thank you.
Okay, thank you so much.
Yeah, good presentation. And actually, what happened that if you plan for this kind of study, practical feasibility comes when you say you have a training, what kind of impact is going to have. Of course, one of the parameters that already includes skills. That is very important. So you have a knowledge, okay, and you can gain the knowledge, but what about the skill? So that can you improve the skill by doing this? So that is one thing is that we have already highlighted the limitation, small sample size, and similar studies have been already done. So it will depend that if it is a bigger sample size, it will be, you'll be able to publish in a good paper. So
Actually,
definitely.
this paper already published in
Yeah.
previous.
So I think you need to go further that how do you have impact of the skill and impact of the training that you need to. And you should have an SOP for that for a hospital.
Yes.
So when you plan for that, you do have an SOP, standard operating procedure, how do you develop a skill? That's how we go. Thank you.
Thank you, Doctor.
Doctor, can I do?
Okay.
This one.
Oh, okay, can.
Yeah.
Sorry,
Actually-
I thought there is no question, sorry.
Actually, you did this study in Saudi Arabia.
Yes.
Do you think the whatever you did
Yeah.
supposed to be can apply in any country?
Yes, actually there was a limitation when I put that I can apply this kind of practice even in Malaysia also. So it will be better because I think we don't have this kind of the policies and also the skill test, which is like more like focused and specific. So this kind of study can be applied in our country or other departments.
Okay, and your conclusion is a higher level of education and high experience. Obviously, it is correct. Any field.
Yes.
It is same.
Yeah.
I think this kind of thing, even you can see the difference between hospital level.
Yes.
Government hospital and private.
Yes.
So that is
Yeah.
Depends on the facility and depends on, of course, experience is better. Sometimes nurse is better than doctor.
Yes, sure.
Yeah. So I think this is expected. Yeah. Whatever you did.
Yeah.
So maybe as a props, at least you can add something.
I think what he asks it, can you implement in different continents? You work in Saudi Arabia, is it feasible for MAHSA? So that only can be done if you develop a model. So you have a model for developing nation or developed nation. So that can be implemented.
Sure.
So then, thank you.
Sure. Thank you.
Yeah, thank you so much. So we end the oral presentation, the first session. I would like to thank the judging panel, Prof. Vikash and Prof. Subhash. And I will provide the microphone to Mr. Rari for announcement.
Thank you, Dr. Savla. Okay, ladies and gentlemen, that concludes the oral presentation. Again, thank you so much for the chairperson, the judges, and the presenters. Okay, now we'll be having a short lunch break, which will be until 2. Our MAHSA University cafeteria downstairs, we have a
in mango. Very sweet. The only thing that I think they want that they don't allow you to plant outside only.
And this afternoon. Moving on with our event, we will have our second keynote speech of the day. I would like to welcome Chairperson Professor Dr. Anam Renita to introduce the keynote speaker and to chair the session.
I'm Assistant Professor working in the Department of Science and Technology. I welcome you all for the second section of MAHSA International Medical Technology Conference on multidisciplinary approaches for sustainable health. I thank the Board of Management of MAHSA and the organizers. Audible? Good afternoon all. Myself, Professor Anam Renita, working in Satyabama Institute of Science and Technology. Welcome you all to the second session of MAHSA International Medical Technology Conference on multidisciplinary approaches for sustainable health. I thank the Board of Management and the organizers for this opportunity. I extend a warm welcome to the keynote speaker, Professor Dr. Subhash Gopinath, who is working as a professor in the Faculty of Chemical Engineering and Technology and Institute of Nanoelectronic Engineering, University Malaysia, Perlis. Professor Subhash Gopinath earned his doctorate from the University of Madras in India in January 1999. He was invited as a postdoctoral research fellow at the Institute of Academia Sinica, Taiwan between 1999 to 2003. Following this, he received the Japan Society for the Promotion of Science Fellow Award to advance his research at the National Institute of Advanced Industrial Science and Technology, Japan, and continued as a research fellow. He has been a visiting researcher in many esteemed institutes. His interest is in RNA ligand interactions and non-coding RNAs in multi-rate resistance, and his expertise is nanoaptamer technologies. In the 25 years of his scientific career, he has written 8 patents, including an immune aptamer that has been successfully commercialized internationally by Vacuum Chemicals Japan. His research works are published in many refereed in esteemed scientific journals, and he has 26 book chapters and 3 edited books to his credit. And he has citations of 13,490 and H index of 57. He has got the Excellent Academic Award by the Optimal Society of Singapore and Distinguished Visitor Brain Gain Malaysia by MOSTI. Further, he is engaging other services as Chief Scientific Consultant in China, Distinguished Professor and Chief Scientist and Adjunct Professor in Malaysia and Indira Gandhi Institute of Engineering Technology in India. And he's a visiting professor in Prince Shokla University, Thailand, Daffodil International University, Bangladesh, and University Science Malaysia. He received the Scholar Visiting Awards in Tianjin University, China, Asian, and Karabakh University, Mevlana Exchange Program, Turkey. Several postdoctors, PhD, and master students from the Waseda University, University of Tsukuba, Japan, University Malaysia, Perlis, University of Malaya, University Technology Petronas, AIMST University, and University Science Malaysia have completed their studies under his supervision. He has many accolades to his credit. I welcome you, sir.
Good afternoon. thank you for the organizers for giving me this opportunity to come first time to MAHSA University. And actually, I am from University Malaysia, Perlis. I think many people know it is in the Perlis state. Perlis is the smallest state in Malaysia, where you can feel like a village. So you don't compare with the KL and Perlis. So if you want to enjoy the retail life, you can come to Perlis. And if you really enjoy the food, you must come to the Perlis. There you can get all kinds of food, including Thai food. So from our place, we can reach the Thailand border and cross the border within 30 minutes. Even we have a campus very near to Thailand. So that is a unique place in Malaysia, but mainly for retirement. Yeah? So kind of less development. We cannot see many industry, but I like it because I'm going to retire. Okay, so usually I start my talk with these two quotes. According to Einstein, the whole of science is nothing more than a refinement of everyday thinking. According to Abdul Kalam, failure can never overtake me if my determination to succeed is strong enough. So both looks sounds like same meaning, but a different thing. According to Einstein, it seems very simple. According to Abdul Kalam, we need to struggle something. It sounds like this. Anyway, so my today's talk is on biomarker imprinting on interdigital electrode for diagnosing human blood clotting disease. So this is our laboratory. So it is in the Paris. So in this talk, I am going to talk about how to imprint the biomarker. What is biomarker? So biomarker is the like a kind of marker that will reveal the disease. For example, recent years, last few years, we had COVID. So we take the COVID test. Usually for COVID, we use the spike protein on the surface of virus, like a biomarker. So another example is if we have a fever, temperature is the marker. Actually, temperature is not biomarker, there is a marker. Similarly, if COVID means there's some protein must reflect the disease. Okay, that is the biomarker. Why we want to imprint? So usually to analyze any molecule, we use the real molecule. But there are a technique, you can imprint the molecule safe. Like you can print the molecule. So that is the technique I'm going to discuss. And in our project, we use the interdigital electrode. That is a kind of biosensor. What is a biosensor? Biosensor is any sensing system that can find your disease. Even for COVID, we have the strip and antigen test. So similarly, that is also kind of biosensor. And we use this system for analyzing blood clotting defect. So this is one of the more successful projects in my laboratory. Okay, next please. Okay, why we need molecular imprinting or biomarker imprinting? So just now I says biosensor is the system that will detect the disease. If you see the any biosensor, for example, there is one here. This biosensor developed in my laboratory. So in the biosensor, basically we have some receptor molecule. What is the receptor molecule? If you want to detect the spike protein, something to receive the spike protein. Generally, people use the antibody. So similarly, we use aptamer. Later, I will explain what is aptamer. And we can use some other protein like a receptor. That is a general system working any detection system. Whenever you go to the hospital, even influenza, you are checking. They must have some receiver or receptor molecule in the system that will receive the virus or the protein of the virus. Then it will detect. The main problem is biofouling. What is biofouling? Biofouling is the non-specific binding. For example, if you are going to test the COVID, but you should not, the system should not detect the influenza. That means one person has some lung disease. It can be influenza or spike COVID or any SARS or MERS, anything. But it should specifically detect particular disease. So if it is not detecting, it detects everything, that is called biofouling. So if you have biofouling in any system, then the problem is it will affect all this system like selectivity, sensitivity, stability, reproducibility. So these four are key elements to develop any biosensor. So for example, you are checking the COVID, if the sensitivity is not good, it is not successful. But even though sensitivity is not good, some biosensors are more stable. For example, the COVID, you have to wait until you reach your temperature high. You cannot check by next day. Your symptom must be very high. That means you must have more virus count. So it depends. But anyway, sensitivity is one of the important thing. Selectivity. It should select only spike protein or particular virus or particular disease causing biomarker. And must be stable. And must be reproducible. Like if I have some sensor, if someone want to follow, must be reproduced. So these all four will be affected by this. So then there are many ways. One of the way is molecular imprinting. What is molecular imprinting? Sorry. Can you go back? So this is the normal biosensor. So we have the simple system. We attach antibody or some other DNA or aptamer. We attach our target. So this is currently people are using. But in our system, we printed. So completely covered with polymer. So that means it will overcome all these problems. Then generally, if we overcome all this problem, that means if biosensor is working good, we call it high performance biosensor. So if you want to do high performance biosensor, you're supposed to use similar one like a molecular imprinting. Yeah? Next please. Okay, so what is our purpose of this project? How we developed it? How we achieved it? So usually, when we cut our hand or by accident we cut, then blood is coming out. After a few seconds or few minutes, it will be clot. But some people, once cut, it never clot, keep on coming. We call it a royal disease, hemophilia. So it is because of the blood clotting defect. So why it is coming? So usually this is the process. When we cut our hand, blood will come out, it will clot. But to achieve this thing, we have to undergo so many conversions. So this is called blood clotting pathway. There are two pathways in the blood clotting system. One is intrinsic and another one is extrinsic. So this is normally happening in any animal system. Okay. So if you reach until here, we need to maintain all reaction. Nearly 30 proteins are involved. So one convert into another. Finally, it will be like a thrombin, fibrogen, fibrin. Then it will be clot, stable clot. So if you have the problem, then it never clot, then an issue. Some accident, keep on coming, people will die, irrespective of accident. Because of blood clotting problem, it will cause the problem. Okay, then what we did, we selected the protein called 9A or 9. So this is called factor 9A, the protein called factor 9. This is factor 11, factor 12, factor 8. So if we see the factor 9A, it involved in both pathway, intrinsic and extrinsic. So then we take this as a target, like biomarker, we develop the system. Next please. So why we need this kind of system? What will happen when you have the clotting problem? So the blood clotting problem always associated with so many problems, like heart failure, stroke, deep vein thrombosis, and pulmonary embolism. So if we take the survey, so there are so many people, nearly 50%, 40 to 50% people will be affected by cardiovascular disease. So that means the clotting defect also one of the factors closely related with the heart disease. Yes. Okay. So what happening? What we did? Actually, as I said, this is the pathway. We targeted on 9, factor 9. So usually what we do to control the system is, for example, I'm doing the operation, open heart surgery. We open the skin, then blood will clot. It is naturally when we cut, blood will clot. So when open, it will clot, then it lead to death. Then what people do? People inject the chemical called heparin. So because the medical people they know. Before operate, they inject the heparin. When open, it will keep like not clotting. So after like finish the operation, it will back to the normal after some time. The main problem with the heparin is it is a chemical, so it will give the side effect. And some people need more dose of heparin, some people need less dose heparin. And another problem is after operation, we cannot quickly stop the reaction of heparin. It takes time. So then we find some alternate molecule that is called the aptamer. This one. Aptamer is a nucleic acid, nothing but either DNA or RNA. Okay, in this case we use the RNA. So what it happened is this RNA will bind to the EGF1 and EGF2 domains of factor IX protein. So if you take any protein, it is the basic structure with the different domains. So if you control one domain, the total protein activity will be suppressed. That is the idea here. So this aptamer will attach this domain, it will be suppressed. Then you can replace the heparin here. You can replace the heparin into aptamer. They tested in the pig already. These people, Reed et al., they are working for several years, nearly 20 years. They tested recently with the pig. So then another advantage is compared to heparin, aptamer or nucleic acid is non-immunogenic. Even you inject into the body, nothing will happen. Usually something enter into our body try to protect. It will secrete the antibody or something. That is the immunogenicity. So if you inject the aptamer, nothing will happen. That is good. No side effect. Another good thing is after you inject into the body, if you want to stop maybe after operation, you can inject the complementary sequence of this aptamer. Usually, DNA, A pair with T. There are 4 bases in the DNA. A pair with T. In the case of RNA, A pair with U. G pairs with C. C pairs with G. So they know the complement. Then they inject the complement sequence into the body, suddenly stop. So because of these advantages, we take this as a molecule and we develop our system. Okay. So what is the mechanism of molecular imprinting? So the molecular imprinting kind of chemical reaction. We must use the polymer. So we have a polymer. We have to do some reaction to imprint the protein. So what we do is, here A is the protein, maybe spike protein. Here factor IX. Okay. So we use the functional monomer, X and Y. We also use another functional monomer, aptamer. So next one. So to make the polymerization reaction, we should do this kind of initiation. I will explain what is the functional monomer. Once we start the reaction, it will make the non-covalent binding. These 3 molecules will be bind here. Usually non-covalent bond is very weak. So then we have to undergo process of polymerization. So when we do the polymerization, the non-covalent become covalent. Then it will be more stronger. So then finally we remove the protein. So here we attach the protein and make the polymerization, then remove. When you remove, there is a imprinting. So usually any molecule, biomolecule, protein, DNA, it will have the secondary or tertiary structure. So once you make this, the tertiary structure will be printed. Then we remove it. So once you remove it, protein will be gone. But the imprint is there. The whatever protein has a structure will be there. So if you attach same protein into the polymer, the particular protein go and sit there because perfect size. So already we made the imprinting, it will come, the same protein will come and attach. So anyway, we undergo 3 process, complexation and polymerization, template removal. So this is the whole process we made for imprinting. Next please. Okay. So another important thing is, as I said, we can imprint anything, DNA, RNA, amino acid, peptide, protein, bacteria, virus, drugs, anything you can imprint. The main important thing is whenever you make imprinted, you also make non-imprint without your target molecule. So this morning we discussed any experiment must have a particular and perfect control. Okay, here one of the controls is non-imprinting. That means no protein. So that is one of the control we must use. Then we can evaluate our system. Next please. Okay. So as I said, there is a polymerization or imprinting. You can do it in the tube and attach on any surface, or you can directly attach on any surface. For example, you have any sensor or chip. Usually we call it chip On the chip, you can directly polymerize, directly attach. There are different methods like surface crafting, soft lithography, electropolymerization. In my lab, we tried with electropolymerization because my lab is engineering lab. We have a mini electrical system. So then we choose this one. So that means there are many methods are available. You can just follow it. Next, please. Okay, this is the overall image, how you can prepare the poly-imprinting kind of a polymerization. As I said, there are different molecules like cross-linker, radical functional monomer, and initiator. So you must have some initiator, then only the reaction will initiate. Then we need to heat up at more than 60 degrees. But the important thing is during heating, we never add protein. If you add protein with heating, it will be denatured. Then it will also collapse the structure under no active. And once we heated, then start the reaction. When a little bit cool, we add the factor 9, then we purge with nitrogen. So finally, your aptamer or any molecule will be embedded. So this reaction actually quite long, nearly 1 day. So we run overnight, and the next day we connect. Okay, next please. Okay. So another important thing is whenever you prepare any imprinting, you must make sure really it is imprinted. So there are different characterization methods like optical based and some scanning based, many methods. We followed almost all methods to confirm whether our printing is correct or not. So here we use a UV visible spectrophotometer. I said once attach the protein, we have to remove it. So when we remove one step, you cannot remove. We remove repeated washing. First washing, so much protein will come out. Second washing, it is less. Third, fourth, slow down. That means we are completely removing the imprinted molecule. So usually we measure at 280 nanometer. So 280 nanometer is suitable for protein. If you choose 260, that is for nucleic acid. So that's I think you may know. Okay. So then we also confirmed with scanning electron microscope. So scanning electron microscope is the electron microscope where you can take the images of the particle. So then we can measure the particle size average. We also checked with the XRD. So XRD is the measurement or it is a system to detect the crystalline or amorphous nature of the molecule or any polymer or anything. Okay, next please. So we also use the transmission electron microscope to confirm our polymerization. See here you can see uniformly formed molecule, here collapsed. So this is the negative one, non-imprinted polymer. This is the molecular imprinted polymer. So this kind of things always necessary. Even if you want to go for good publication, high level publication, you should have everything proper. So if you have everything proper, so no question, you can reach anywhere you want with the journal. So we also find the temperature stability, the improvement of stability with temperature we can find. We also analyze the Raman. So Raman usually we find 3 peaks with the carbon material. So the polymer is the carbon. So all all chemical molecule must have the carbon backbone. So we can see the 3 peaks carbon, and we also analyze by EDX with the TM. So you can see carbon and oxygen. The carbon is very high. So because the polymer is highly compact with the backbone with the carbon, and we also use the NMR. So we see we confirm with almost all studies. So this is another thing. Another thing means I said we need to deposit on any system, any chip system. So this is the one of the method we developed. We produced the chip in the laboratory. You can also buy from commercial source, but you can also prepare in the laboratory. In the laboratory, we have a yellow room, light protector room. So we do all these things under light protector room. Basically, we make some patterning. So initially, I say interdigital electrode. Interdigital electrode is the kind of electrode. It has 2 probing system, positive, negative. Any current must be positive, negative. So to pass the first negative, we have the 2 electrode, and we have the cross-linking band size electrodes. So, but here we convert the parallel electrode into the circular one. So that is the flexible. Any design you can do. So, but there are different steps like aluminum etching. This is the sensor name, and this is a wafer, aluminum deposit, photoresist, photolithography, pattern develop. All these things we need to do. to make the complete sensing device. Yeah? Next please. Okay, once we prepare the sensing device, we need to confirm again. So any experiment, you need to confirm all steps. Then only you can establish the lab, you can establish the system. Don't skip anything. So here also we checked with our microscope system. So we have different microscope, 3D nanoprofiler, high power microscope, scanning electron microscope. We check with everyone, all microscope. So we can see very nice pattern and intact uniformity, everything we can see. Then we can go to the next step. Okay. So this is after polymerization. Again, we need to confirm. So that is before polymerization. So we prepare the polymer separately, chip separately. Now we coat it. So when we coat, it will be attached like somewhere in the chip, and we have to check with the electrolyte. So whenever you talk about positive, negative, electrolyte is important. For example, you use a buffer. If you add the buffer, there is electrolyte, positive, negative. If low pH, there is a acidic, high pH, alkali. So that we need to make sure. For example, if you are checking the blood sample, so there is a pH. So pH should not influence whenever you use the electric system. Since we are using electric system, we want to make sure the pH will not influence. So then we check the pH. So you can see at alkali range, there is some unnecessary current process. At low pH and neutral, almost nothing. So this is the process like between positive and negative. So there is a molecular movement. We call it a dipole moment. So if positive, it will move towards the negative region. If negative, it will move to the positive pole. Yeah. So it is like a moving. So we checked with all our system and confirmed. Next please. Okay, this is our system. We call it probe station. Probe station is nothing but only two needle. We pass the current through the ammeter. One needle will pass the positive current. Another one is the negative current. Basically, this is AC current. Yeah? So this is our sensor. Here is the positive, here negative. So here needle, here one needle, here one needle. So we attach here, and initially we checked it. So when we absorb the factor IX protein, if low resistance, there is a high current. If high resistance, there is a low current. So initially the problem we encountered with the molecular imprinting is it is not highly linear, either quickly saturate or it is not perfectly linear. Then what we did is we input some more nanoparticle. So we included two more nanoparticle, single-walled carbon nano horn and gold nano Rg. So this is the gold, this is the carbon. When we include these two molecules, the level of enrichment is very high compared to normal one. So then we find the way how we can improve further. Okay, next please. So this is the system we did. I told you I attached two molecules, two nanoparticles. One is a nano horn, nano Rg, gold nano Rg. So this is the gold, this is the carbon. So now we use two molecules. One is carbon, one is gold. So whenever you design this this is actually a very simple idea, but we managed to publish in good journals. So this journal recently under minor revision, this is in fact 20 plus. Idea is very important. Yeah. So we incorporate different nanomaterial. So we attach one way. We cannot attach one shot. So you need to know if carbon, what is the linker you have to give. If gold, what is the linker? For example, if carbon, you can give the amine. If gold, you have to give the thiol or amine. So you must know some basic chemistry. Then you can attach. So this will enrich the performance of the sensor. Next please. So once we attach, again we check with our system. You see, we have the simple imaging system. Every step we check. When we check, we can see the gold and the carbon. Carbon is coming from the nano origin, nano horn, and the gold is coming from gold nano origin. So there are many systems that will confirm the molecule attachment. Okay, next please. So then we also check it under a microscope, scanning and transmission microscope. We confirm all particle size and the shapes, uniformity, indentness. So we also check it by UV visible And here you can see non-origin. Origin is the sea animal. I think you see this like a lot of thorn. So we can see similar structure. That's why we say gold non-origin. Okay, next please. So when we attach the nanoparticle, we can see the current flow. Here looks very better. Here going very deep. So this kind of thing is very good and good for reaction system. So we confirm this thing and then we proceeded further. Next slide, please. And we analyze the comparison. So whenever you develop any system, you must need some comparison. So here we compared with like a molecular imprinter, non-imprinter, and after sensor. After sensor means only directly attached, conventional. So we compare all three. The blue line is the molecular imprint. It is always better, and we can get a good sensitivity. Actually, factor 9 people usually detect at picomolar level, but in our system we reach the femtomolar level. That means more sensitive. Next please. So as I said, the previously we did in the tube then attach, and we can also directly attach. We use the method called electropolymerization. So electropolymerization, we need something like a 3 electrode system. We have in our laboratory. So we pass the current and add the polymer. We need a different chemical. It is called polypyrrole. So it is a polypyrrole. So it will attach like a layer by layer. Whenever we attach, we can see the response in our measuring system. So that means we confirm each layer and we proceed with the second method. Next. Okay. So upon electropolymerization, again we checked. So we can see more crowded molecule compared to my previous method, and we also checked with FTIR. So FTIR is the system that will find the surface chemical groups. So we also find the clear peaks with FTIR and confirmed with all microscope. Then we proceeded next level. Next please. Okay. So these are all our development in the laboratory. Okay, so we imprinted polymer we developed with a different method, and we optimize, and we go to the good level of sensitivity on integrated electrode. So this is like a completely wet experiment. So usually wet experiment refer to laboratory. So then we also tried with dry methods like in silico analysis, like molecular analysis. This you can do easily. Nowadays, you can go to the database. You can see all data from any molecule, any protein. So we take a molecule from the factor 9 molecule structure from the database, protein database, and we analyze the molecular modeling in silico analysis. We find a different domain because we want to know why our study is correct or not. And that's why, even though we are engineering, we move completely to the structural biology. So that way, my lab achieve usually more. We publish more. We achieve easily. So we bring different evidence. You see, my slides, we have so many evidence. Each and step we give evidence. Then no questions. Wherever we want, we just publish it. Okay. So anyway, structurally we analyze and confirm this is really binding. My aptamer and factor 9 is really binding. Next one. And this is the another structural analysis, like a Ramachandran plot. Plot. So usually Ramachandran plot is I think physics people know very well. Ramachandran is very popular in India. So he is the father of physics. Yeah. So based on a Ramachandran plot, you can analyze the backbone of molecules. So we also confirmed the important binding region, the aptamer CUA, where it is binding exactly to the protein. So we bring all evidences and we confirmed. Okay, next. And we also find the stability. This is also based on in silico analysis. So you can see initially molecule is stable, at the end little bit unstable, because with the time we cannot measure. Even initially we measured it with picosecond. Then journal rejected. You cannot do picosecond, nanosecond. Then we repeated with the nanosecond, but we don't have a facility. We send the sample to India. So someone helped means the commercial. They are doing the outsourcing. So we did it in a nanosecond, then we published. Okay. And then this is another one, we made it like a validation. So you know ELISA. ELISA is one of the very popular technique, gold standard. mainly for AIDS. HIV, they use the ELISA. Then we use the ELISA. ELISA is usually using the antibody. ELISA is the aftermath. So this is an alternate method for ELISA. We also confirmed by ELISA. So it is also some kind of confirmation. And we use the UV visible spectrophotometer. With increasing the protein, you can see the increment in the peak level. Yeah? So this kind of confirmation we accumulated. Actually, this is a PhD student work. My PhD. She is going to have the Viva. So she did all. Okay. Next, please. Okay. So this is our final product. So you see, we started from scratch. We developed our own biosensor. So this is a system like portable. We call it a point of care or bedside analysis. So that means we can use the system at any places. So usually bedside analysis means you can keep it at home. So you can analyze. Point of care, you can take it to any field. You can carry it basically. So nowadays, this kind of sensing system is getting popular. Usually, this system can attach to your mobile phone. So we have apps, we can just connect. So we analyze. So even you can give the link to your doctor, family doctor. You can analyze at home. It will reach the data will reach your doctor. So that is the future direction. You know, nowadays artificial intelligence, IoT, big data, all are booming up. So in future, I think we lose so many people will lose job. All robot will do the operation. All car will move automatically, artificial intelligence. So like this, the medicine also, no hospital, no clinic. So only you call your doctor, the measure, your doctor see your results, and he prescribe your medicine. So you call the pharmacy will bring the medicine to the home, like your home delivery. So the everything will be automated. So we are just touching the direction. So we also like attach the Bluetooth, so we can reach to the telephone. Okay. So then, okay. So this work already patented, operated. So next please. Yeah, next please. So this my work received several best awards. Next please. Please, next please. Next, not be previous, please. So we all please back. Okay, back. Next. No. Sorry. Forward. Next. Yeah, next. No, there is another slide. Go back. Slowly. Next. There is another slide in between. I think this slide is missing. This one? Yeah, this is yeah. So this work also received so many gold awards. I think the organic side. Yeah, okay. So these are all my students. So my PhD student in my lab, you can see all communities, Indian, Chinese, Malay, and we have interest in Nigerian. So I give all opportunities to my students, and she is the only student more aggressive. So far, my best student. Usually we teach the student, but so far I learned everything from her. It is not I teach. I learned. She taught me. So she is the best student. She received all awards. She received all patenting, all everything, complete PhD. And so far, she is the best student. So in the past, I established so many international collaborators. So I have so many collaborators, even 20 years, 25 years. So this person nearly 25 years, here 20 years, 10 years, 20 years. So I maintain the collaboration. And the local also, I have so many collaborators. I have no space to put the photos. So every university, I have a collaboration here in MAHSA. I have Dr. Suresh. So we are collaborating for nearly 10 years. So this is the way we are moving our laboratory. And so our student usually very aggressive. Almost all students, they finish their graduate on time. They receive the best thesis award every year. So when finish the PhD within 2 years, they will reach cumulative impact at least 40. So only this girl reached more than 50. So that means we work aggressively. We managed to maintain so many collaborators. So managed to submit at least 100 papers per year, ISC paper, not conference. So we submit at least 100 paper ISC papers, and so far succeeded. And next slide, please. Next. So I end again with initially I started with the 2 quotes. Now I end with 1 quote. The whole of science is nothing but more than a refinement of everyday thinking. So if you ask me, I support Einstein. Science is very simple. Just to think more, do less. So usually we think some gaps in the literature, we work on this and publish in the good journal. We never publish in journal paper. So this is the way we are running. We are so far serving to the science, and we are also surveying. Thank you for your attention. So if you have any question, you can ask me, and we can discuss anytime. Usually I available 24/7. Midnight I reply to my WhatsApp. Even my laboratory open 24/7. So you can enter anytime. If you want to collaborate, you can visit the police. No charge. So we have many facilities. I know data sources. So you can send your student, or you can collaborate. You can come. You are always welcome. Only thing is we cannot support for your accommodation, traveling, but laboratory completely free, 20%.
Hello, doctor. I just want to ask, because I'm not sure about your machine. Like cabbage patient, I know that deficiency of factor 7, they will bleed. So we are 7. So for example, can you use your machine to detect whether they have deficiency factor 7, and also for those extracorporeal mechanical device, they are checking factor 10, especially when they receive heparin. So your device can detect only the clotting, or is it any deficiency, so that that we no need to give factor 7 in case they are bleeding. So I'm not very clear, doctor.
Actually, this is a correct question. Not only my sensor, any sensor, unless the specific one for COVID or something, this kind of sensing system is generally only the chip we always change. It's like a cassette. If you want to particular song, you play with a different cassette, like this. So if you want to detect, you use a molecule suitable to capture the factor 9. If you want to check factor 7, you can attach the aptamer or antibody specific to factor 7. Similarly, thrombin is okay. So that's why one of the recent development is multiplex detection. In my system, now I use only one chip. We are making now 3 or 4 chips. One chip we attach factor 9, one chip factor 7, one chip thrombin. Like she said, we can detect anything. Similarly, for lung disease, we can attach COVID, influenza, SARS, MERS, so whatever you want. So even TB. So like this, we are developing that is called the multiplex. It is highly possible with this system. Actually, this system is common. Only the chip you need to change. Which one you need? Yeah.
I wanted to ask, like, why you chose factor 9 in printing?
Yeah.
Is there any to choosing?
Yeah,
choosing
the
that?
2 reasons actually. So if you see the blood factor 9 involved in the 2 pathways. Actually, we have 2 pathways, intrinsic and extrinsic. There are 30 proteins in the clotting pathway. If some protein coming under intrinsic, it will not be in the extrinsic. If coming under extrinsic, it will not be in the intrinsic. But the factor 9 is a common one. It will involve in the both pathways. That is the one thing. And another thing is, as I said, there are 30 protein. Factor 9 coming at the initial stage. Any disease we must predict from the initial stage. So that means early detection. One is early detection, another one is it involved in the 2 pathways. Then that's the reason we choose factor 9 as the biomarker.
So do you have any other ongoing projects which detect factor 8, factor 7 and all?
Actually, our laboratory is kind of everything like a setting up. If you want to initiate factor 8, you can just start straight away. You can start. That means already we have all set up. So our laboratory is like kind of we are like we are going to hospital. You cannot say only this disease. Any disease you treat. Like this we have machine. If you want to come to our laboratory, whatever you want to detect, we can make arrangement on the surface of our sensor. So it is not depends on new project. It is not like any project. You can straight away you can do any project. The basic system is basic platform is important. We have a basic platform. You can use our platform for any application.
Just one last question.
Any question?
Is there any what is the clinical utility? Like what diseases can be monitoring of factor 9 levels? Like so hemophilia it will be helpful in detecting and monitoring hemophilia or any other diseases?
Hemophilia is one of the one, and like a heart attack also. For heart attack, we have many biomarker like troponin, C-reactive protein. Factor 9 also one of the biomarkers for heart attack. So since it is in the bloodstream, it is related to many diseases. I remember it is also related to some brain disease. So if you take any disease like brain disease, like Alzheimer, there is a particular biomarker is important, but the factor 9 may be the secondary biomarker. So if you find some differences in the factor 9, you can also predict it may also affect the brain. Something like it is like a second, you can say secondary biomarker because it is in the bloodstream all over the body. So it is like a bloodstream. So we can relate to many diseases, but major diseases is clotting.
I'll ask one question. You use the different molecules to have the imprint, right? Including one of the aptamer.
Yes, actually, aptamer is not necessary for imprinting. So only your target is enough. But in this project, we also include the aptamer because when you bind with molecule, the structure will be more perfect. Yeah, that's why we include aptamer. So
there could be other possibility that we can have multiple aptamer from different domains of the protein. We can have the imprint only with the aptamers.
No. Like
A, B, C.
No, if you imprint with only aptamer, only aptamer can bind. No, this system is a little bit different. Yeah, you are right. Generally, if you put the aptamer, some other molecule will bind. But here, that's what I said. No need aptamer. The target must be imprinted.
Yeah.
To hold the target, we put the aptamer actually. So here, what you say, different aptamer, not different aptamer, different protein, like a spike protein, influenza protein, like TB protein, something like this.
And same imprint, but instead of different molecules which can bind to the different domains, we can have the different aptamers which can bind to the different domains of the same protein.
Oh, that is the extent of this work we can. But I think there is a possibility.
Yeah.
So.
Okay, thank you.
Yes, thank you.
Thank you, Professor.
Thank you.
Thank you, Professor Dr. Anna Marita, and also thank you, Dr. Ratana. It was an informative speech and it was wonderful. Next, we are going to move on to our next segment, which is oral presentation session, which will be held in the auditorium. And also, we'll be having poster presentation session, which will be held outside of the auditorium. I would like to request all the poster presenters to be at the respective posters. The judges will be judging the posters soon. I hope all the poster presenters are at their respective place.
And after that, this call.
Now we'll be having our oral presentation, and the judges will be Professor Dr. Rusli Nuruddin and Associate Professor Chin Jin Han. And also, I would like to welcome Dr. Sudha Armugam to introduce the oral presenters and to chair the session.
Thank you, Ms. Suzanne. And good afternoon, everyone. Welcome to the second session of the oral presentation. So just to lay down the rules for the session. So each presentation will be for 10 minutes and 5 minutes for Q&A, right? So we will have two bells. First bell at 7 minutes, that is just to alert you. And the second bell will ring at 10 minutes. So when you will have to wrap up. And the next 5 minutes will be for the Q&A. Okay, so I once again welcome our judges, Professor Dr. Rusli bin Nuruddin and Associate Professor Chin Jin Han. Okay, so without any further delay, we will start our oral presentation. Okay, so I welcome our first presenter, Ms. Vanaja Perumal. Okay, sorry for the confusion. So I welcome Dr. Kin Dudley Thun. Her title of her presentation is The Significance of Microsatellite Instability and Loss of Heterozygosity and its Association with Clinical Pathological Parameters in Invasive Ductal Carcinoma of Breast at Yangon General Hospital, Myanmar.
Good afternoon, the respected my dean and my colleagues and my students. So this is the title. So regarding about the about the microsatellite instability for loss of the heterogeneity of the gene in carcinoma of the breast. So IDC. So breast cancer is the most frequently occurring cancer in the public health problem as well. So this is an introduction slide for it has been predicted the worldwide incidence of the breast cancer is almost number 3. So very dangerous. So it should be aware of this is the American Cancer Society as well. So ITC means as invasive ductal carcinoma, the most common type of the breast cancer. So among the 75% is according to the American Cancer Society, about 75% of all breast cancer is ITC, invasive ductal carcinoma. So there's I also kept it in the reference as well. So what is MSI? So microsatellite instability is called the short arm is short tender repeats. This also we call is the simple sequence repeats SSR. So this is a short repeats of the DNA sequences. So here see the microsatellite is here. This is a normal, goes a normal and the replication. If something has mutation, deletion, or the mutation has happened, the test will be here. Is there something has abnormality? So this result will be after the RT-PCR real time of the reverse transcriptase polymerase chain reaction. So there's a MSI will be detected. So it's found most commonly in the breast cancer, adenocarcinoma, and colorectal carcinoma, and the CA stomach, CA endometrium, those type of cancer, including breast cancer. Usually we can check there's either MSI or LOH. So microsatellite instability or the loss of the heterogeneity. So this is here. So I would like to explain about the LOH is here. This copy A and copy B. The DNA has the paternal side and the maternal side. So here is see in the copy B, this is missing here. So this is the instability of the gene. In case of LOH, the one copy is lost. This is called mutation. So this is a study objective to find out the primary invasive ductal breast cancer case in that hospital and categorize the histological grading. So we used to follow the Blue and Richardson classifications and to detect the breast cancer related MSI and LOH by using the SDR. We extract out the 7 DNA and we using the primer. If the metastasis involving the lymph node, we take it out the 7 DNA. So to find out the association between microsatellite instability and loss of the heterogeneity clinical pathological parameter. So research has a mixed method approach, both quantitative and qualitative, apply in this study to explore value of the MSI and a multiplex PCR analysis detecting the genetic instability of these 30 cases of the patient. So study design, molecular pathology, hospital-based and laboratory-based. Sample size is only 30 cases. The key equipment used are histopathological lab, H&E staining, DNA extraction, paraffin abundant tissue, and the PCR and page analysis and gel documentation. So this is a 6 loci what we have used and 6 microsatellitic marker for chromosome. So selected were these 6, and then if there's a metastasis, we use another one more. So I would like to explain about my students as what is the genes and the nucleotides. So the C, G, A, and then it may be no. So the genetic informations are stored in the DNA. So DNA only then copy rather than the RNA. So the background is amplification techniques for the cloning specific and target plant parts of the DNA sequence to generate the thousands or millions of the copy after we extract it. So the letter has no meaning like the alphabet. So genetic is alphabet and then only words become the meaning. So this is a negative charge. Genetic information will be sequence. So genetic information are stored in the DNA. So nucleotides, let's say, is the letter. Gene will be the sentence, and chromosome will be the paragraph, and genome is the book, like our telephone book. So the human genome has 3 million nucleotides. So we will extract out the only one loci, Alice. So when it can be multiplied, one amplification. So PCR will need 3 main steps: so denaturing, annealing, and the extension. Each and every step we need a primer. So set 6 mean we need 6 primers. So it can be specified for the each specific gene. So it's based on the, we have to check for the temperature as well. So annealing, and with the primer, it will go extend. So then replicate. So where's to start, where's the end? So where's the gene has extracted and which segment has mutated, we have to check. So it can be duplicate copy. When we do the amplification, the potential amplification will be go cycle by cycle until 34 billions. So it's logarithmic multiplication. So microcytolytic stress of the DNA sequence will the single nucleotides or 1 or 2 units of the mononucleotides is repeated in the genome. So we also mark with the fluorescent marker, then we can check with the color. So the result is the value of the microcytolytic, the multiplex PCR analysis detecting the genetic instability in that cancer. So this is what I have explained about those things. So this is a result categorized as a compared with the clinical pathological parameter, age, and the tumor size of blue and richardson, grading, presence or not nodes, and the metastasize, and the grading, and the menopause, perimenopause, and postmenopausal status. So prevalence is a 32. Then the which genes? The chromosome number 5q32 is the mostly 25 cases is found, and other LOH has loci is chromosome number 7q and 21 is found. So this is a percentage distribution of the microcytolytic instability. So analysis of the high polymery microcytolytic instability loci not only provide the information about the instability, but also allow detection of the alleles, deletion or mutation. And then the Z displays the high degree of the heterogeneity at the cytogenetic level, so detected by the methods of that comparative genomic hybridization. So altogether, within 3 years, 30 cases. So this study, the surgical breast biopsy are histologically diagnosed, IDC, then ever increasing number of the marker of the value, ensure that validates the published study as well. So these are the procedure method. What we use is the PAP method, is a peroxidase and peroxidase technique of immunohistochemistry method. So these are the keywords. So this is a reference. Thanks.
Thank you, Dr. Gandhi, for finishing on time. So I pass it on to the judges for the Q&A.
Yeah, thank you, Lali. I just want to ask you, because basically this is, I don't know how to call it, is it actually a study or just, because basically, you know, when we look at the MSI, for instance, in a way that, does it allow for some prognostic, what do you call this, value? Because you're saying looking at the instability and heterozygosity, you know,
Yes.
so whether it is actually it has diagnostic or even prognostic. I'm not sure which is the one you are targeting now. Is it diagnostic?
Diagnostic is very
Yeah.
sure, definitely diagnostic.
Yeah.
But prognostic value is it would we have a classification, and prognosis value is based on the ERPR oncogenes. So it will direct treatment site. Prognosis factor is based on the age and the which site and types of breast cancer. So it's not much related to the prognosis factor. It's not much
Okay,
significant.
the question is that basically now in terms of screening for breast cancer, you know, now you see intraductal carcinoma. What other form of breast cancer?
Can special type
Yeah,
also can be.
because I think now the important thing now in most countries, the first thing is screening. You must screen because many women didn't come. By the time, like in Malaysia, for instance, by the time they come to the medical attention, it's too late. Already stage 3 or 4, many died from breast cancer. There are survivors, I know. So critical now is that this method of looking at cancer, is it? Just I say, is it? Can it be used more as a screening tool? No? Now say diagnostic, but screening to maybe screen people for not only IDC but other type of breast cancer. This I think is important. Yeah.
Because we will know the chromosome sequence only. What is happening in the gene? So based on the type of the breast cancer only, it's related to the prognosis. So this is for the treatment.
Okay, thank you.
Is it goes to chemotherapy?
Thank you, Dr. Tali. So I make to understand that your study is conducted in Yangon General Hospital, Myanmar. So why you choose that hospital and when this project is done?
My thesis. And why I choose is I work there. So easily accessible. We have to communicate with the surgeon, and then also the H&E stay. The process are so many steps. So it's a one-shot service for me. No easy, no need to travel anywhere.
So it would be good if you can include the ethical approval reference number in your slide.
But we need only the block.
Let's say now when you have finished this project, what would be your next plan?
My plan is I would like to focus on the molecular rather than the tissue level. Nowadays, we should think molecular, at least minimum PCR. PCR is everywhere, even they freely given. PCR, the molecular gene, we must, all our eyes, hairs, everything, skin color is come from the DNA. So molecular, think molecular.
In your study, you put 30 patients, right? How do you select these 30 patients?
30 patient is who has a confirmed ITS diagnosis using tamoxifen. So that patients are available, and we just collected the block, paraffin block already.
Thank you.
Thank you, Dr. Dali. Okay, so now I call upon the next presenter, Mr. Eric Tan Min Zuan. The title of his study would be Impact of Phosphate Solubilizing Bacteria Inoculation on Plant Growth and Secondary Metabolite Production in Mexican Mint, a comparative study with different fertilizers. Over to you.
Good evening, all. So, my name is Eric. Okay, good evening, all. My name is Eric. So today I'm going to present my project, my research project that I have done last time. So in this research project, I was doing about using the bacteria, especially using the phosphate solubilizing bacteria to apply on the plant and see how the plant grows. Together with the different fertilizers. So the Mexican mints that I use this plant as in our project is because that this plant, the Mexican mint, the Pentanthus abonicus, this plant that, as we know that this plant is a herbaceous perennials plants that have medicine properties. It can, in the folk medicine, they use to cure fevers, flu, constipation, and also asthma, and also skin disease. So during last time, the people that use this plant for this kind of used for medicines properties. And because they'll have the phytochemical compound, which is flavonoid, ester, and also aldehyde, which can use these properties to doing some use as a medicines. Okay. So in this project, I was doing like, we will see the phosphorus, because the phosphorus is the micronutrients. They are naturally they're present in the soil, which is an essential nutrients that help plants to grow. So the function for the phosphorus, they have photosynthesis, DNA and RNA synthesis, and also the energy transfer, help cell division and plant growth, and also improve the stress intolerance for the plants. Okay, so for the objective in this research project is that we're going to apply this bacteria in our plant, which is the Mexican minks, in different fertilizer conditions. So what is our specific objective to do this? Is that we are going to isolate this bacteria from the mother plant of the Mexican minks. So we get the nature's bacteria. Then after that, we will put into our plant to grow. Then we also evaluate the qualitative of solubilizations efficiency and compare the qualitative determines the available phosphorus in between wheat or wheat consortia. And we also have doing some tests, like we have using with or without the consortia. So we're going to test the chlorophyll's content, as well as others like antioxidants properties and antimicrobial activities. And also we have squeezed others like the total phenolic and flavonoid content in Mexican minks. So this is the workflow starting from I begin for this research project. So starting for the seedling stage, there I use the plant, the Mexican mint. I cut into half. I cut into in the small part, like the second picture they show. Then after this, I place into the seedling tray to let it grow first. This is the first step, and after this, I go around to search those mother plants of the Mexican mint and get the soil from them. And in this time, I collect the rhizophyll soil and also non-rhizophyll soil to get the bacteria. And after this, after I have get the soil, then I select the bacteria. Then I will test the phosphate solubilization's efficiency. So in this test, I have using the triple superphosphate, ROS phosphate, and also the vermicompost as my different fertilizers conditions. So we will also have to test whether it can give more phosphorus contents. It can release more phosphorus. Then after this, we prepare the consortia. So after the seedling tray, they grow they come out with some leaf. Then we will transfer to the bigger box into the group. Then starting from day one, we will starting to put the consortia, the bacteria. Then in between, every 15 days, we will do some plant parameters. And after this, we will do the phosphate solubilizations estimation, similar to like the efficiency test, but for this test, we will use this to test the phosphorus content, but we have mixed with the tricalcium phosphate. Then after this, we have to do the test of chlorophyll estimation. After we do the chlorophyll estimation, we will start doing the plant extractions. So in this plant extraction, we are using soft slag extraction method to extract the content from the plant. Then after this, we will use this plant extract to run the test, the antioxidant and antimicrobial test. Okay, this is the first one. Then after this, after it start growing from the seed tray, then we will transfer into this pot. So one pot got 6 plants. So total I have 8 groups. 4 groups is without consortia, another 4 is with consortia. And this is the group, no fertilizers, without the consortia, then rosophate, triple superphosphate, and vermicompost. And then this is the result. This is I isolate the bacteria from the soil. And this is the highlight one is it contain highest bacteria. It can give a better yield for the plant. So this 4 is the one that I choose to use as a consortia to put into the plant. Then this is the phosphate solubilizing estimation. As we can see, the TCP, which is the highest one, the consortia with TCSP, with TCP, they have a highest phosphate at the 72 hours, then getting decreased in 96. So this is we see the phosphorus, because all these fertilizers we are going to put in our environment group, also the experiment group. So we'll see how the phosphate content, how it can produce, how much they can produce. Then this is the plant parameters. We also can see the phosphates, rock phosphate have a better height of the plant compared to other trees. Then after this, number of leaf. Also the rock phosphate, especially the experiment group, have the highest one. And this is the chlorophyll estimation, which in this chlorophyll estimation, the experiment group, which is the rock phosphate and the triple superphosphate, have the highest chlorophyll contents in the plant compared to the environment group without the consortia. Then this is the plant extraction that I have done. Then this is the amount that I have get from that plant with or without the consortia, and this is the amount that I get the plant extract to further do the test. And this is the test that I have done, the DBPH test. And we can see the triple superphosphate have the highest one, together with the same diagram, the phenolic and also the carbonate showed triple superphosphate have the highest. Then it's the bacteria, the antimicrobial sensitivity test. They were using the test Klebsiella and also the E. coli and also the Staphylococcus aureus. We also got the positive controller they were using, and we applied in C, and this is the result, and show that in the experiment group they have a better antimicrobial property against to the Staphylococcus aureus. So this is our last one. So in this test, we are still going to do this plant parameters and also the antibiotic sensitivity test, also the minimum inhibitory concentration. So in future, we also hope that for those students who, if they are continue to do this test, the soil analysis, the PSA and also chemist tests can be done in future. So in conclude, so in the experiment group, with phosphate solubilizing bacteria, it showed in our result, it showed that they have a better use than compared to environmental group, especially in phosphate, also the triple phosphate. So we hope that in future the soil analysis and also can help to analysis the exact concentration of micronutrients present in the soil in order to get more specific data to compare. These are all my presentations.
Thank you, Eric. So now I pass on to the judges for the Q&A.
Thank you, Eric.
All right.
Although some of the things define my understanding how you do it, but just a curiosity, asking you from a different angle. Why was in your study the focus on phosphate? Again, I said asking you from that angle, because the understanding is that phosphate is the main macronutrient in soil, and therefore you're targeting phosphate using this bacteria, consortia, to improve, am I right? The property so that the plant become the best in terms of growth, and later on you did soil testing, yeah. But besides phosphate, is there any other macronutrient that normally you look at the fertilizer environment that could contribute to this? Because it seems that the focus only on phosphate. Are you thinking that phosphate is the only thing in the soil?
Yeah, the phosphate is the main, but they also got other nutrients like the potassium, also one of the micronutrients. They're very important for the plant growth. But in why we choose this phosphorus is because actually we're supposed to do phosphorus and potassium, so we have better data can compare, but due to the time limitations, so we only do the phosphorus.
So basically you are saying that there are many other macronutrients besides phosphorus, potassium, the other one could be other things, other maybe even magnesium. I don't know. There could be some other macronutrient. Maybe because I ask it because what we don't want is some issue about it could be not just a single factor that was contributing. It could be multiple factors, multiple macronutrients acting together and producing the desired effect. So that is my thinking. And secondly, this in terms of the what call this utility of this experiment that you're doing, how does it helps later on in terms of promoting this method of what call it soil fertility to derive maximum yield of the plant, and then also to because you try to prove that it has very high antimicrobial activity in it. Because you are ongoing work now, you are doing MIC, minimum inhibitory concentration you are doing, am I right? You are still ongoing work, yeah. So because this is important, this the antimicrobial activity is an important aspect of this research. So you try to, and then the other one was chlorophyll count you mentioned, and then antimicrobial. So basically that the a few outcomes that you are looking at with a single experiment. Okay. Are you very sure that this purely because of this effect? This is my point, no? Because it seems that this single activity of the what call this consortia acting on the phosphorus solubilizing bacteria, and then you derive all this outcome. Is that what you're trying to conclude?
Research experiment, there we are going to see the consortia to affect to the plant. So if let's say this consortia can help the plant to have a better growth, because in even the TSP, they were using the fertilizer, we're using the rosophate, we're using the triple superphosphate. These are, we call as, we still need to further to, we have to use this bacteria to further to degrade this fertilizer into the soluble one, soluble phosphorus, to let the plant have a better growth. So once the plant can get a better growth, so it means that it can have more healthy grow, like increase this chlorophyll's contents, and also improve the plant abilities, like it can have a better antimicrobial activities against this bacteria.
My last question is that why you choose mescaline? Why the other plant in Malaysia? No, there's many plants, huh? That they equally are in folklore herbal tradition. And they say very powerful antibacterial. So the question is, why are you choosing mescaline?
Why I'm choosing this? Okay. Because they have others. Because in MAHSA, they have at the first actually I choose is the okra, but like the chili, the okra, they got others people already done. But the mescaline is no one have done before for this plant. So this is the reason I choose. But I know because this plant they doesn't have the fruits. So if you using some plant with the fruit, you can have a better data to do the compare. But mostly the plant they already have choose by others. So that's why I choose this mescaline. Because mescaline also in previously they also have using as a medicine, not only cure some disease, not actually in the disease. They also used to use as to cure this constipation, some skin disease or fevers. So this is the reason I choose this one.
Okay, Eric, in your conclusion, you say that the BSP grow better than the other group, right? How do you come to that conclusion? How do you
PSP.
Yeah, how do you analyze your data?
Okay, because we can see if we compare to this.
Yeah, what test do you use to analyze the data here?
Using the DPH, using this DPH test, the antioxidants assay, to see which group they have a better activities for this one. Then we can see at the triple superphosphate, the yellow bar, is a experimental group that with the consortia and with the TSP. Compared to others group, it have a better percentage compared to others. And not only this, then we can see the TPC and also TFC. Because we know that the TPC and TFC, they are together with this. Also, these two tests also important in antioxidants. And it showed that both of these tests also showing the highest level, higher percentage on TSP on experimental group. So then we can see the chlorophyll's estimation also at the highest. Yeah. So in my conclusion, then we can conclude that the PSP it really help plant to grow better. But if they come with the TSP, it can let the plant grow much better.
Thank you, Eric. So now I call upon the next presenter.
amide, which is an amide isolated from Melastoma malabatricum leaves. So what are my contents? The contents of this presentation are these things. So why I have chosen wound healing activity is that because of the global wound care. Okay, wound. Okay, global wound care, which is the cost of the global wound care in 2014. If you see, it is around 2.8 billions now that per year, and that it is now projected that in 2024 the global cost of wound care yearly would be 28 millions, 22 millions by 2024. So because of this, it is very interesting for the academicians or the researchers to work upon and research upon these wound healing products or methods and technologies for the betterment, whatever may be the source of the wound, the cause of the wound. So, so basically, what is this Melastoma malabatricum? It belongs to the family Melastomaceae, and there are the plants belonging to this Melastomaceae. These are usually found in the tropical and subtropical regions, which include Malaysia as well as the Indonesia, as well as the tropical countries like Cambodia also, and that there are around 12 species of Melastoma species which are available in Malaysia, and that it is traditionally used in wound healing besides that dysentery, diarrhea, and ulcers. So the main focus of this study is to find the molecular mechanism involved in the wound healing activity of auronamide, which is isolated from Melastoma malabatricum extract in primary murine fibroblasts. So, research question: how auronamides do exhibit wound healing properties? And what is the research objective is to determine the molecular mechanism involved in the wound healing properties of this auronamide. Then hypothesis, null hypothesis: the wound healing activity of auronamide is not mediated by the expression of the genes associated with the with this wound healing, which involves the hemostasis, inflammation, then neovascularization or the granulation, reepithelialization, and remodeling. These are all the different steps involved in the wound healing. And that the other then regarding this overall methodology, what is the thing is that the plant material, the leaves are being bought in Sungai. It is authenticated by the taxonomists, and specimen is also provided to them, and then UPM, and then it is air dried, and then it is extracted using Soxhlet apparatus, using different solvents like a hexane, ethyl acetate, methanol, and that we have conducted this the isolated flash chromatography we have used to isolate the auronamide, which is an amide which is isolated from Melastoma malabatricum leaves. So, and that we have conducted this cell culture work, and that we have done the wound healing activity, especially the migration test, as well as we have used the scratch assay test for finding out this wound healing activity assay, and migration test is also conducted, and that we have tried to find out the gene expression using the qPCR. This is our overall picture. Then isolation of this how we have isolated. First of all, we have taken 250 grams of the leaves of the dried leaves of this Melastoma malabatricum, and that we have extracted, and that ethyl acetate extract was concentrated. Of course, we have done the extraction with different solvents as mentioned, and then we have our main extract that we have we could manage to isolate this auronamide is through the ethyl acetate extract, Then around the extract that is being produced is almost, the extract the yield is almost 27 weight per weight. Next, the isolation of auronamide. So flash chromatography, Bucci flash chromatographic technique, when that the column silica gel is the extract is being solubilized in methanol, and that it is chromatographed using silica gel, and that the mobile phase are hexane ethyl acetate methanol, and that the auronamide which is obtained. There are many other components also, but the auronamide which is obtained is characterized using this NMR, UV, IR, NMR, as well as mass spectroscopy. Then, so this is the NMR of auronamide, 13C as well. This is LCMS auronamide peak. So the HPLC, the peak is found, that is the molecular weight of this eluted peak is around 5006, and that it is around eluted at 6.6 minutes, 6 point, okay. And then next, cytotoxic studies. So what are these cell lines that are used for this study is that 3T3 cell lines are used, and that this auronamide is dissolved in DMSO solution, and that we have used 0.05 DMSO test solution, and that to dissolve this one, and that because it is a universal solvent, and that the maximum. So we have used the blank as well as control in this one. What is the purpose of this cytotoxic study is that to find out which concentration is toxic to the humans, or to find out that toxicity levels. We have done this. Next, then this is how we have done the formula with which we have calculated. Then, so we have found that the even 1000 micrograms per ml is also safe. So next, so wound healing studies. So we have to find out first of all whether it is safe or not. We have found through the MTT assay, just no colorimetric test. We have done the cytotoxic test, and that we have identified that it is even 1000 micrograms is also per ml is also non-toxic, and that wound closure and migration. These are the formula with which we have it is calculated, and that we have found that auronamide this helps in the wound closure in a dose as well as time dependent way. So it is doing even all the 0 and 10 nanomoles as well as 100 and 1000 we have done. And then this is the wound closure. So you can see that 48 hours total closure can be seen, and 24 hours it is started. And then qPCR for the quantification of the gene expression, we have done this one. Then GADPH gene was used as a reference gene. Then these are my results. These are my study. And then what is the conclusion is that this auronamide is helpful in wound healing property, and that this it is having a potent wound healing activity at 100 nanomoles per nanomoles by regulating what are the genes that are being regulated are these CCL2 as well as CCL10, CXL11 as well as VCAM. All these genes are being expressed, and that besides that there is these show that this is the mechanism with which the auronamide is showing wound healing property. So this is regarding this. So this project basically it is an FRGS project which was given for MAHSA University, and that the student Law Junction has actually graduated with this FRGS project which is given to us, and the one who has supervised closely with them is Prof. Mallikarjun Rao as well as Dr. Kavita Mohandas, previous colleague, and Dr. Tikki. And then next, these are my references.
Thank you, Dr. Madhu. Now, Prof. Rusli.
Thank you, Madhu. I've got just one or two questions. The first one is that basically this very basic question. Number one is the plant itself. You mentioned that the plant that you're using for the study. Who actually confirmed that that's the
UPM taxonomist. Taxonomist
yeah.
from UPM.
So there's someone to be sure that it's the plant that you're using. And then number two, because the study it seems that you're using crude extract. Crude extract, Marana?
We have
yeah.
this project
Crude
is
extract,
for
sorry.
the project,
Yeah.
the crude extract, but we have isolated.
Yeah, this is the thing. So my question is this, first you have crude extract, and then you isolate the aronomite. Okay? Be isolated. So this aronomite, and then you were able to detect that the level at which it has this activity. So the question is that if you have already extracted aronomite, could it be not only from this plant? No? Could be even derived from other plant. I don't know.
Yeah,
And,
it is already derived component.
so the question is, if you could have isolated the aronomite, can you use the aronomite alone to show it is there is an effect on wound healing? Yeah? And then the next one I want to ask you also, this wound healing, you're doing it on animal study or what? Wound healing.
Wound healing.
Where's the wound?
In vitro study.
Huh?
This is all in vitro study.
In vitro study. Yeah. So how do you create the wound? Because see the thing that
So we have found the genes which are expressed during wound healing. We have studied for that.
No. My question is that how do you create that wound? The wound can be example. Example, no. Example is the diabetic wound. The classic now we are so worried now about people with diabetes. No, they have diabetic wound. If we could use say aronomite, apply it say onto that wound, and then because now you already showed acceleration in terms of the healing property, like what do you call this? The wound you showed by a series of activity in it until wound closure and then complete healing. So that which will be a fantastic what do you call this finding, because if you can apply even just a diabetic wound, I think at the moment you're right, because the wound industry is a $1 billion industry. People are looking for something. Even now, we are looking for any medication to accelerate wound healing. So now you have identified aronomite, and if it can be identified, it could even be done on say Tumira on diabetic wound and see what happens, rather than only in vitro or in vitro kind of activity. So I understand this the phase of the study. You have not gone to clinical trial. You just at this level still using it in vitro and so on, or even animal study. So what is now my question is that what is your plan, long term plan towards like even clinical trial and so on?
So at the moment we have first of all we have identified whether the plant is what is the mechanism of the plant, how it is what is the mechanism of the plant in wound healing, and then we have found and isolated, and that we have tried to identify and understand this one. So maybe later clinicians may take over that to the higher level. Mine is a fundamental research.
Okay. So you have completed this project. What is your next plan?
There is we can go further for this because which are the compounds? There are few other compounds are also having this wound healing activities. We can go for further studies for these compounds that were isolated and having a wound healing component.
Okay. Has it been tested in the animal study through your projects about the effect of this on the wound healing?
Animals we did not.
Okay, so what do you think? Let's say if you do this study in the animal study, what is the difference of the in vivo study compared with your in vitro study?
This one is more like homogeneity is there, and that you can have a better results are more better. But that individual, if it is an animal, the study is a bit not as that of the. But later we have to go for different studies.
This the last one. Basically,
any plan to do some toxicity study, because
Toxicity studies, like we have done the MTTSA work, cytotoxicity test we have done.
You've done it.
So we have found that it is safe until 1000 moles per, that one is per ml is also it is quite safe. That's why we have done for the procedure.
Thank you, Dr. Madhu. So now I call upon the last presenter for the day, Surya. She'll be presenting on the title Organic Fertilizer in Food Production for Sustainable Agriculture Development in India.
Good afternoon, everyone gathered here. This is Surya from Vivekananda College of Arts and Science for Women Autonomous. And first of all, I need to thank everyone for this giving this wonderful opportunity. And I need to thank to my management and to my parents. Because of them, I'm just here. And now, the title of my presentation is Organic Fertilizer in Food Production for Sustainable Agriculture Development in India. So as the title itself will tell what I'm just going to do. Then the next one is, so this was the thing which makes me to do this project. According to the United Nations Population Fund, the population in India is increasing, not only in India, over the world, it is increasing up to 2.6 billion people. The current data was this. And especially in India, the population is about which is 1,406 billion population. So due to overpopulation, most of the farmers are forced to use the chemical fertilizers and pesticides to increase the yield of the plant. So to reduce the fertilizer product, to reduce the chemical fertilizers, I'm just going with this project. And the next one is objective of the hydroponics. So here we are including the technique which is hydroponic. So it is not a new technique, it is an ancient technique which has been followed in Japan and some more developing countries over the world. So this is a technique where the plant have been grown without the use of the soil. We are growing the plant completely in the water. So we are just mixing the nutrition what the plant was needed. So we just mixing the nutrition in the water itself. And then next one. So and according to this technology, I have correlated with the organic fertilizers, which means the organic fertilizers will help the people for their health benefits also. And so we can here what I have done means I have reduced the chemical fertilizers and reused the waste into the best pesticides and as a fertilizers. So when we are consuming the organic food means the health benefit for the people will also increase. So we can get more antioxidant content while consuming these organic food items. Next. So for the study, I have selected the Solanaceae family. Why I have selected the Solanaceae family means, so this was the most common plant varieties what we are using in India. So without this, there is no food in India, which is like Solanum tuberosum, then Solanum melongena, then Solanum lycopersicum. So this was the most commonly economically valuable plant. So only I have selected the Solanaceae family for my study. And next one is I have selected the two plants. So that's which is chili, which is Solanum, chili and the tomato. So next one is I have selected sample 1 and sample 2. So for that I have analyzed the water. Why means this is a technology we have completely growing the plant in water. So we need to check whether the water is quality or not. So I have tested 3 tests. One is easy electric conduction and the total dissolved salt, which is TDS, and total hardness of the salt. And next one is this was the methodology what I have done. I have gone with 2 types of system. One is wick system and deep water culture. Based on the plant morphology, we have selected this type of system. And this was the plant growth over stage by stage. And next one is for the sample 1, I have used the vermicompost. So vermicompost is nothing but which is waste which has been produced from the earthworm. So that which may contains like macro and micronutrients what the plant was needed. So I have taken the clean vermicompost and soaked in water for 4 days. After that, I have filtrated removed the debris and then I have just used the liquid fertilizer in a diluted formulation. And next one is, so after applying the fertilizer, which is vermicompost fertilizer to the sample 1, we can see the growth of the plant. And next one is, so after that it shows the flowering and fruiting for the sample 1. So and simultaneously I have also done the soil culture also. So I have compared the study in both plant also, in soil and water plant. And next one is, so this was the final stage of the plant. So and next one. So the result of the sample one is chili needs 6 hours of direct sunlight and the pH has been maintained based upon their based upon the TDS of the water. And then it taken 20 days after transplanted into, it taken flowering from 20 days after transplanting to deep water culture, and then it taken 15 days to become a mature plant. So I have analyzed this result based comparing the soil study of the same plant, and when comparing to the soil plant, this shows the best result. And next one is sample 2. So for the sample 2, I have gone with the deep water culture. So this is nothing but a tomato plant. And next one is for this sample 2, I have used the fish amino acid. So how I have prepared the FAM means I have taken the waste fish from the market and then I have mixed it with the brown sugar and then mixed them in an equal proportion, and it was fermented for 22 days. After the fermentation, I have just taken the liquid compound and then I have diluted in the water and I have used for the plant. And you can see the picture of the plant next slide. So after applying the FAA, you can see the growth of the plant. Next. So from this you can see the some part of the plant have been affected by the insect. For the insect affected also I used the organic pesticide to remove the insect. And next one is, so this was the sample 2 result. So as well I am comparing to the soil. So this shows the best result. The next one. So from this I have concluded that when comparing the chemical fertilizers, so organic fertilizer would be a good one for the human health. And also when we are growing the plant in soil, it takes more time to grow, and the yield of the plant also would be get later because the root need to search for the nutrition in the soil, and then the growth stage will be took long more time. So when we are growing the plant in water means we can directly mixing the nutrition in the water, so it is more easier to uptake the nutrition from the water itself. So in the future perspective of the studies, so we can use different bioformulations like the waste material which is from we have taken from kitchen waste or from agriculture waste, and also we can mix with some microbes like vesicular or vascular mycorrhiza. So in future study, I am focusing on that. And also we can grow with the some more crop plants like rice, which is mostly using one of the stable food for the India, so that in future may trying that also. Thank you.
Thank you, Surya, for giving a very fast presentation. I think everybody is hungry also. So now over to the Q&A.
Thank you. This is important study because what hydroponics. Now we also have the aeroponics now. Huh?
Yes.
So many phonic now. Yeah.
Yes.
So many rather than the soil phonic. Yeah. Yeah. So my question basically is that I mean this again Dr. has been doing this under eco-village. This hydroponic study become a new trend of hydroponic. So my question is basically simple, no? Because it is so what do you call this important to have organic food. No? You mentioned
Yes,
free from pesticide. We are trying to remove pesticide, purely organic, and it seems that it is easy to do, easy to plant. It's quick to get, and especially in the current economic climate, where food is very expensive now.
Yes.
Now in Malaysia, now you go outside, no, people are now there is a decrease in number of people are willing to eat outside. Very expensive food. We go even China also, Turkey China also cost how many ringgit, no? So eventually, that the question is that when you talk about hydroponics, you're talking about economic, because people now must be able to produce their own food. In the end, really, you really have to be self-sustainable. Everyone, you have to produce your own food, good food, organic, free from pesticide, and then you can sustain and it's sustainable, So on that note, so you are now applying to some of these crops like chili, this common chili, tomato, So in the long run, we need also other staple food that are common in Asia, like you saw rice, Rice so common, yeah. So in terms of the of this what do you call this I don't know to call it experiment, more like now it's a venture to create more awareness and to have more hydroponic. So what is your take on later not only in India, even Malaysia, is there a possibility of doing hydroponic on the big scale, especially in urban community, people living in the high-rise flat, condos, they can. I saw people even planting, and so it become a new approach towards nutrition, nutrition needs of community. So what is your thinking? Can this hydroponic as well as aeroponic, is the next generation of food production in the world?
Yeah, surely.
What is it?
Yes, sir, of course. In future, because due to increasing population, the place for the three needs to survive in this world is food, shelter, and the food. And
Jika udah orang makan.
It is yes, correct. The same name, mainly you call it this only. Yeah.
So as I said, so in future that would be the issue, which means the space consumption. So from this we can do the vertical farming. So vertical farming done means the space consumption also reduced, and the faster need of the food also will be done by using this technology. But mostly the developed countries are following this technology, but some more countries also need to do this technology because for the future need. But surely it would be possible, I think so.
Can we propose to government, politician, that is the next thing to get voted in into office?
Yes.
They have to support
Yes,
scheme.
yeah.
Sure. It can be turned to a political tool. You want to be in office, you want to encourage and encourage. Get the paisa, support all this hydroponic activity.
Sure.
Yeah. Please, Anyway, the one is political.
Thanks.
Okay. Can you just share with us what is your understanding about when someone says it's organic food? What is the criteria to be considered or categorized as organic food?
Yeah. It's a good question, sir.
Because in your objective you say to create something organic, right? Just ask.
Yes, sir. So when we are consuming chemical fertilizers, it surely affect our health. Why it's organic fertilizer is important means our ancient peoples have just followed the same thing, which is they have gone with the organic fertilizers, so they lived healthy based upon their what you have seen in the past. We are just following the present and the future. So if we follow the same as like ancient people means, you know that in our ancient peoples are survived up to 100 years because of their healthy food. So now we are just staying up to only for 40 or 50 only due to uptaking of
40, 50 is very less.
Due to
We are
uptaking
already
of-
40, 50.
Yeah, sir. And
You are
most,
scaring us.
and most of the people are affected by diabetes, heart attack because of their basically started from the food. So just following ancient people. So as we are seeing our past people, we just need to go with the future. That's it.
Okay. Let's say based on what you say is correct. The main objective is to get rid or avoid for like pesticide, right? From these like chemicals, this and that. So now based on your setting in your project, how you ensure the water that you give, the soil that you use are free from the pesticide and the chemical fertilizer?
So soil is so that soil have been not used chemical fertilizer because I have taken from field only.
No, water.
Water is
What sir is saying what?
water is normal tap water only. That would not that may not mix with chemicals, I think.
So you assume that the tap water is clean.
Yeah.
Is that so?
Yes.
So basically, sir, just want to ask that only. Have you done any kind of testing, soil testing or water testing, so to ensure that that thing
Yeah,
that you are
okay.
in is pure?
But I have not done it, sir.
That's what sir is saying.
Yeah.
You should include these things.
Okay.
Prof is saying that.
One thing, actually, they check the heavy metals.
Oh.
Generally, heavy metals.
Soil. Also in water.
Yeah. So as sir told, you have to check for the product heavy metals. Organic means less heavy metals. And just a suggestion, if you add any plant hormones, whether you can get more yield. Generally.
natural plant hormones.
Natural plant hormones.
Yeah, sir, it may be possible.
You didn't check.
Yeah, I have not checked, sir.
So just on that note,
Maybe aeroponic is much more superior than hydroponic.
Yes, sir.
Because you know why? Aeroponic does not depend on water. No? What do you think?
That need more less water than compared to hydroponic.
Yeah. So I saw there are many projects now using not hydroponic anymore. It's aeroponic. They say it's superior. I don't know.
Yes.
There could be a better study to compare, no? Which is the better one in terms of in the organicness of it, free from heavy metal, pesticide, and so on, and then the yield. Because now the population around the world is talking about this more food and cheap, and easy to construct in their place.
Yes, sir. Hydroponic is more better than the Thank you.
Thank you, Surya. So now we have come to the end of the session and the end of the day one of my Med conference also. I think Betty is ready in level 2 spine. I think all of us can go down there to level 2. And I request you all to come back tomorrow